A very big pleasure to have all of you together now for this follow-up symposium so early after the first symposium. And I would like to say that that is a particularly important one, because here we probably have something that is important beyond sarcoma, even probably beyond oncology, because the fundamental issues we hope to tackle here are shared with great many very rare diseases and the ways to find medicines for that. Now, let me first very shortly focus on the issue we do have here with the assessment of marketing authorization for these very rare diseases for promising products. We all know that the classical gold standard, statistically significant results in 2 randomized controlled trials with clinically relevant endpoints are never going to happen yet. But we do want to find way forwards for this promising new treatments that could be there. And that is why we need, from the regulator side here, to ask ourselves, do we have the right tools? Do we need the tools we have to use in a different way? Do we have to find and create new tools for the regulatory decisions? Because we do want, let me repeat that, we do want to find ways to make this promising new approaches, promising new substances, promising new treatments available. And when I say promising new treatments, that immediately leads to another challenge. Promising is, of course, something that for patients, for their physicians, is something they would like to have the chance to use. But for decision makers in the regulatory and other fields, that is usually not the evidence we want. Promising usually means that is a justification for a clinical trial authorization. And that is a justification for someone to invest, to finance here. And that is another thing we need to tackle today, we want to tackle today, beyond the regulatory issue. Even if it is promising and under circumstances that would interest someone to invest, for very rare diseases, it might be difficult to find someone who is willing to go there. And no blame here. Let's be realistic. For companies, they do have a legal obligation to their shareholders that is definitely bigger, stronger than their obligation to the stakeholders. That is not blaming. That is just, dear friends, being realistic, expecting an investment in something where no return on investment is expected. Not even a chance for that. A decent chance. That is something that is simply out of the question. You can even say, hey, come on, is that embezzlement? So what we want to do today is going beyond, importantly, dear colleagues, dear friends, going beyond just identifying problems. What I do really hope to get from this symposium is that we start drafting at least possible solutions, possible ways forward, designing from scratch, or looking at positive examples that can be used and say, hey, look, there is something that can be copied, that can be modulated to show a way forward in this admittedly difficult area. So therefore, no further delay from my side. Again, very, very happy to have you here. Very happy that we managed to have that in such relatively short time after the first symposium. Big thanks to the people behind it. Caroline, Francesco, big thanks. And I leave then to Pierre to structure what we do today to have a very productive outcome, to have tangible results. Pierre, burden on you.
Thanks a lot, Harald. So welcome to anyone. Thanks a lot for engaging into this dialogue, because my my main word today will be dialogue. I believe that if we want to make things possible, it will be after exchanges, not only exchanges just to talk. We have many things to do, but because these talks could result in making things progressing, to make medicines and progress available to our patients, because it is our patients who matter here. So what can we offer at the level of the EMA? I will have a short talk at the end of the first session to explain what could be, in my opinion, speaking on my own behalf, the role of all stakeholders. Stakeholders here, I will mainly mention the academy, the physicians, the patients, and the regulators. The regulators at the level of the marketing authorization, because I'm not responsible for making approved drugs available to patients. But we are speaking mainly of drugs that are already approved or not that expensive. So would it be a problem? I don't think so, not necessarily. And we are speaking of an amount of money which is necessarily limited by the limited number of patients. So I believe that the financial aspects of this, from a public point of view, are relatively negligible when compared to the importance of the question. So welcome, because we started discussing together, and I hope I will bring some preliminary good news. The EMA is really open to that type of discussions. I even had an opportunity to discuss briefly through emails with the General Director of the EMA, Emma Cook, who gave me her strong support for this initiative, saying that it was very important to have these questions discussed between you collectively and us. So welcome. This is the second part. I believe, I not only hope, but I believe that at the end of this day, we will go back home, not necessarily with a solution for each development. We are not that magic, but with good solutions for organizing everything to make our support, because I believe really that EMA must be supportive within the limits of our regulatory role. So to organize our support to make it as efficient as and as available as possible. And this is something which is important, being available to open and efficient to make difficult things becoming possible. So this is my short introduction without any slides. And once more, a very warm welcome to all of you, because discussions with people like you are very important if we at the EMA want to progress and do our job even better. This is my motto, my personal motto, I would say. Thanks a lot. Thanks a lot indeed. And let's have this meeting.
So I also want, on behalf of the EORTC, and particularly the EORTC Soft Tissue and Bone Sarcoma Group, to thank first EMA, Pierre, Caroline, Francesco in particular, to be open to meeting us so often and to try to discuss together about this very relevant point for us, that is how can we improve the access to active drugs for patients with ultra-rare sarcomas, as an example for all patients affected by ultra-rare tumors. Today here we have representatives from several, let's say, partners. In addition to the EORTC, we have patients, many patients, I would say, and these patients are from the sarcoma community, but again, they want to be here to represent all rare cancer patients. Then we have colleagues from other countries outside Europe, and I really want to thank them for joining, because we know that if we want to improve in ultra-rare tumors, we need to go globally. Otherwise, it's not possible to gather the data that are needed and to conduct studies, and even not possible to attract companies. So we really believe that it's relevant to coordinate this effort with our colleagues from other regulatory agencies. This is very important to us. And then we have also a representative from non-for-profit organization, and in particular anti-cancer fund. And this is important to notice because we really need to work together to make progresses. This said, I am very much aligned with what Pierre was saying. So we are here to try to refine how we can improve the opportunities for discussing together the challenges, the issues that we are facing every day. And this is not the right place to discuss of a particular drug or of a particular disease, but still today we will make some concrete examples because our daily life is done of examples, and these are the good opportunity for us to also enlarge, to open the discussion to other condition, other situation. And I want to, to conclude before introducing Pan by saying that this kind of a workshop of discussion was let's say, suggested following the pilot project of repurposing. This pilot project that was conducted by the EORTC Soft Tissue and Bone Sarcoma Group, Anti-Cancer Fund, and patient was the example for us of how discussing together, meeting together, trying to understand each other point of view and also challenges is the only way to identify solution for the problem we have. So we are here for European citizens, for worldwide citizens, for our patients. And I again want to thank everyone for taking time to join and to use our brain and our heart to improve what we can do. This said, I don't know if Pierre wants to introduce maybe Pan.
Yes. So Pam will summarize what has been concluded after the first part, and probably will open for the discussions we should have today. So thanks a lot. It's a very important talk because it's organizing the follow-up discussion. Thanks a lot for that, Pam.
Thank you, Pierre, and thank you, Sylvia, and thank you to the EMA for enabling us to do this. We realise it's incredibly important for everybody. So what I'm going to attempt to do is summarise in a few short slides a whole day's discussion. So you can imagine that that's quite a task. So I apologise if people may have misrepresented or misremembered certain points, but I think I've captured, based on the notes that we took and the videos that we took, I think I've captured the essence of the day. Areas of agreement. We all agree, I think, that although this is under the umbrella of ultra-rare sarcomas, these are an exemplar of the problems that we face for all ultra-rare cancers in general. So we're not here to talk about specific histologies or specific subtypes of ultra-rare sarcomas. We're looking at problems that affect all ultra-rare cancers. We all agree that there's an urgent need to address the issues that are a consequence mainly of the low patient numbers in these ultra-rare cancers. And these are things like difficulty in accruing patients in clinical trials. You may only have a few patients. It makes randomization problematic. It makes international working essential because you can't do it in a single country. A consequence of this is also generally a lack of interest from commercial drug developers. The rarity of these conditions means that there are gaps in our knowledge of the natural history of the disease. And we have huge gaps in the knowledge of the biology of the disease, especially in histologies where we have a very high phenotypic and genotypic heterogeneity. I think there was generally agreement that the regulatory pathways that exist, which have evolved over the years, generally for conditions where there are more patients. So the more common cancers, the more common diseases, where we have greater uncertainty, there are also attendant problems. I think there was general agreement that the clinicians and academics who work in the ultra-rare sarcoma space may be less familiar with regulatory requirements than colleagues working in cancers where there's a huge amount of regulatory activity with new drugs coming online and so on. I think there was an agreement that programs such as Accelerate can help in bringing together all the key stakeholders, the regulators, patients, clinicians, and pharma to collaborate together and speed things up. And that's essentially missing from the ultra-rare cancer space. And I think there was also a recognition that we are living through a time when there are massive changes underway in the new pharma regulation. And some of those articles, like Article 48, 164, 84, articles on real world data and things like that, are going to have a significant impact across all diseases, but significant in our diseases as well. So I think they were the areas where we had consensus, but there are also challenges that came up. And a key challenge is, given the nature of the diseases that we're dealing with, we have to learn to integrate non-traditional data streams into regulatory decision making without compromising the very high regulatory standards that exist. Nobody's asking to lower the bar. Nobody's asking to change things, but we have to recognize that the gold standard randomized controlled trial is an extremely difficult undertaking for these diseases. So we have to learn to integrate all of those things, like prospective registries, innovative trial designs. And we heard from people during the first workshop about some of those ideas. We need to learn to integrate those. We need to learn how to strengthen the non-randomized data collection that is going is being done at the moment. We need to learn how to strengthen single-arm trials when they happen. So we need to work more closely with trialists and regulators to kind of get the most value from the activities that we're doing. And then finally, I think there's an agreement that we need to build a collaborative environment, something that's more agile, something that involves regular meetings, co-creation of an environment that's conducive to change. And one example of that might be the push platform that was also mentioned on the day. So this is a snapshot of the agenda for today. It's structured around those questions. And I highlight here the fact that we've got discussion sessions and that we should use these discussion sessions during the day to really kind of thrash out how we want to solve things. I don't think we can solve things in a meeting like this, but it's important that we understand how to work together to solve them. And I think that's a key deliverable. So let me finish with what I think is a really important statement, that there are really high unmet patient needs, and those needs demand that we act together to effect change. And we want to do this quickly and rigorously. And I emphasize that word rigor because it's very important that everybody is comfortable that we come up with solutions that maintain standards, but also recognize the situation that we have in front of us. Thank you.
So thank you very much, Pan. I think now we move to the next session. And I leave this to Ralph Farrell from EMA and Winan Vanud from the EORTC to present, to introduce the next speakers. Thank you.
Thank you very much, all. Very excited to discuss together with you for a second time and address what Pan just outlined. How do we understand to get to solutions together? So we have in the next session now 4 presentations support an ecosystem for ultra-rare cancers from diagnosis to treatment. And I'm very excited to first have 3 presentations that come from colleagues. And then I heard from Pierre, you will present what you announced in the beginning, suggestions for what the development could look like. So great pleasure to invite Hugh to start off then with the patient involvement, hospital cohorts, and mobilizing the patient community. Thank you, Hugh, for also today presenting and sharing how this comes into the ecosystem. Thank you.
I wanted to start, as Pan did, by thanking both the EMA and EORTC for organizing this event and for giving the chance for patients to have a voice at it. We think patients have a huge amount to offer, as I hope I can show, but it's not always the case that patients are fully involved. So thank you very much once again for that opportunity. Today I'm going to talk about the patient involvement, and we're looking at this from 2 aspects: hospital cohorts and mobilizing the patient community. And I'm going to start with hospital cohorts. So if we could go to the next slide, please. Here's a model, and I want to repeat something that several people have said here. I'm in an organization that is disease specific, but today I'm talking on behalf not just of ultra rare sarcomas or ultra rare cancers, but I think as Pierre said, any ultra rare disease will face many of the same challenges. If we look at a simplistic model, what I call the individualist model of how ultra rare diseases might be looked after, I've tried to depict here 3 countries with multiple centers of care at hospitals. And what we see, I think, generally is that without patient coordination, etc., where the only groups are the patient cohorts, patients tend to remain extremely isolated. What we see also is that in terms of the standard of care, and I don't mean the quality of care in terms of empathy and etc., but I'm talking about procedures here, there are significant differences between countries about how a disease is handled. And even within the same country, between hospitals, we see differences in how diseases are treated. That means that scans, types of scans, regularity of scans, blood samples, a whole range of different ways that the disease is monitored can be different, and the data can be stored in different ways. And what that translates into is data that when you try and collate it into a study group or something has a lot of variation. And I think that is why many people, and certainly regulators, are quite nervous about engaging with retrospective data. So the question is there a better way of doing this, or could there be? Let's go to the next slide, please. I've called this the dream question mark. This is where exactly the same hospitals now are all following exactly the same protocol if we're treating a disease. And obviously a protocol, how a disease is treated when it's heterogeneous, will depend on what's being faced. But if every hospital were to be following exactly the same protocol and storing exactly the same data in exactly the same way, then we get much closer to a sort of prospective study where data could be trusted. We also like the idea of having centers of excellence in countries where, for ultra rare diseases, the data and information of every patient is collated in one place to build as big a picture as we can of that disease. But then those centers of excellence would have an obligation, we think, to feed back to the hospitals their experience and understanding so that we can collectively grow the understanding of the disease. Very often what we find is that hospitals may have no patients with an ultra rare disease, or they may have 1 or 2. So there's not a great incentive for them necessarily to try and drive to the dream, because their focus, of course, will be on far greater numbers of the common diseases. So this is just a challenge. It's not a criticism. It's just a challenge we face with ultra rare diseases. The question is, can we do anything about this? Can we go to the next slide, please? Can we transition from the individualist model to what I might call the coordinated model? And I think, yes, we can. But to do that, all the stakeholders involved need to be working towards and with common goals to deliver better treatment and better outcomes for the patient community. And patients have a huge part to play in this. They are hugely invested in finding the results and moving the understanding of the disease forward for obvious reasons. Now, I am going to get slightly more specific here because I want to just talk about the EHE model as an example of what we've achieved, because it may be useful. So if we could go to the next slide. In 2012, the EHE community globally was in the individualist model. Patients were not coordinated at all. There was one small registry started in Australia by Sylvia Pollock, but that was all that there was. There was no funding, no patient voice, and really little attention being paid to the disease, except by the doctors who were dealing with it in each hospital. In 2013, something quite special happened, and that was that a few women in America found each other, found the support they gave each other amazing, and just wondered if there were other patients out there who might also want that support and join in. And so they set up a Facebook page. And that Facebook page started to grow very quickly, not just in America, but what they found was that they had patients joining it from all over the globe, because no country anywhere in the world had an EHE patient community. And so as it grew, the excitement grew and the voice grew. But what we then realized was that that was fantastic for patient support. But in terms of moving our understanding of the disease forward, it wasn't going to make a huge difference. To do that, what we needed to do was drive a proactive research program. Research means you need money. Money means you've got to raise it. And to raise it, you need properly formed foundations, etc., with all the tax benefits of donation that come with it. And so in 2015, the EHE community Facebook page set up its first three foundations, one in America, the EHE Foundation, the EHE Rare Cancer Charity in the UK, and the EHE Rare Cancer Foundation in Australia. At this stage, we'd gone from 5 to 500 members on the Facebook page, growing all the time. We were amazed at the speed with which the fundraising took off. And in fact, 8 months after we set up the UK charity, we were able to donate $180,000 to Brian Rubin to fund his lab, who were at that time the only lab we knew of that was specifically looking at EHE. It's not just money. Setting up these charities, the trustees often don't know what they're doing in terms of medical diseases, etc. Patient communities, they need support. So we need to set up advisory boards with clinicians in who do know what they're talking about. And so we start a dialogue between patients and clinicians, and then researchers come to the party because they begin to hear about the activity. We have funding to drive research. Today, on the right-hand side, we now have a Canadian entity, and fantastically, EHE Italia has also been set up, went live at the end of last year. We have today over 2,600 members worldwide. They're not all patients. A lot of those are supporters as well, but we have hundreds of patients from 85 different countries. We've raised over $10 million so far, and we've been able to have, I think, a fairly serious impact on the research picture. If we go to the next slide, please. In 2015, when we decided we needed to raise money for research, this was our understanding of dedicated research groups who were dedicated to EHE in the world. And I apologize to anyone here who, I'm not saying this is true, this was our understanding. I'm sure there were people possibly around who were also working on EHE, but this was the one dedicated team we knew. So did we have an impact? If we go to the next slide. That's the picture today. We have 26 major projects going on. Some of them have actually, we did a PhD in Manchester, that's now finished. But we have projects ongoing. And I think that I've just put that up. Don't try and read, it's too small what the different projects are. But it's just a visual indication of what can be achieved when you mobilize the patient community. And that's not just the research. It's not just research. It's the conversation that takes place with clinicians. It's the awareness of the disease. It's the ability to get involved in serious advocacy programs, and actually to come along and talk today about what we've achieved. But as I'm saying, this is just our example of what we've been able to do with patient mobilization. We're not the only one. The Chordoma Foundation have done a fantastic job, and I'm sure there are a lot of other rare diseases who are doing a similar thing. If we go to the next slide, please. This is just a quick list of some of the achievements that we've contributed to, either through the funding that we've been able to provide or by mobilizing patients and engaging with our patients to support the information. We helped identify the actual fusion proteins that drive the main two forms of EHE. The TEAD binding that was identified and is now a major focus of drug development. Our Manchester PhD has identified DNA damage and damage to the homologous recombination pathway as a model that might explain why we see indolent disease and why we might see it go to becoming more progressive. So that's really interesting because it is today, as far as I know, the only model yet that potentially explains why we see this change in aggressiveness to progressive, etc., in EHE. In 2015, there were no models for EHE of any sort. Today, we have Brian Rubin's robust GEM model, which is fantastic. We have a stem cell model in Manchester. We have human cell lines being developed in the US, 2 or 3 of them. We have a zebrafish model being developed in Sheffield. And also today, not on here because we didn't contribute to it, we have the PDX model that was developed in INT in Italy, which was fantastic. The research we're funding has also helped and is helping to identify biomarkers, and I think GDF15 will be talked about today. That study has also developed a fairly major observational study with 50 patients now enrolled between INT and the Marsden, and we're hoping over the next 3 years to increase that to 100 patients. We have also funded or part funded the setting up of the first ever prospective multi-center prospective registry of EHE, which has 22 hospitals in Europe participating. And I think at the end of last year had the first patients coming into that study. So that's fantastic. We have biobanks now funded and set up in America and in the UK. These are really critical for rare diseases. Losing tissue and losing samples is heartbreaking. And so having biobanks set up where we can capture this is really important. It's a huge learning curve when you start these things, because when someone first talked to me about biobanking, I said, okay, well, where is the UK Rare Disease Biobank? Who runs it, etc.? And everyone looked at me and said, well, there isn't one. So, we have to, and again, not a criticism, this is what the patient communities of rare diseases deal with. We're not moaning about it. We're dealing with it, and we're trying to fund what needs to be done. We've also set up the Global EHE Patient Registry, which now has over 200 people engaged with it, entering data, etc. And we're beginning, and Denise will later today be talking a little bit and showing some data from that. And I mentioned right at the beginning that it's not all about research and funding, but we've also been able to assist clinical trials in getting patients to engage. And the Mechanism trial in America was a good example of that, where we were able to ultimately, I think, achieve over 40 patients involved in that clinical trial. If I had to describe all of this or attribute one word to it, my word would be momentum. Because what happens is that in this sort of program of activity, you see momentum building, momentum in interest in the disease. In 2013-14, if we went to ASCO or CETOS, EHE probably wouldn't even be mentioned. Whereas now we go, there are discussions about it, there are posters about it, etc. So it's really great to see that happening. Okay, if we can go to the next slide, please. So the patient communities are a really powerful partner in anything you do. If it's globally coordinated, mobilized, and as I've said, hugely motivated. But it does have some reasonable expectations. The patients want to be fully involved and they want their voice to be genuinely heard. I can assure you they will not be ignored. They want all parties to work together to try and find solutions so that things like common practices developed and as a result of dealing with common diseases do not become an impediment to progress for rare diseases, no matter how comfortable we may feel using those established procedures. They also don't want to see or hear that the rarity of their disease or the lack of profit is a major impediment. They want solutions to be found, and I think those can be found. Ultimately, what they really want also is that all of this happens with appropriate urgency. And urgency is a really important word for patients who have these diseases. Rare diseases are not rare to the people who have them. They're very frightening. And I would just ask everyone, whenever we are meeting, whatever we're doing, whatever we're looking at, ask ourselves the question, how does this bring forward getting a new treatment to patients? How do we accelerate the process? Because that is really, really important. Okay, if we go to the next slide, please. So obviously engaging with patients is fantastic, but what we also want to do is optimize that engagement. We can't afford to waste time or lose opportunity or not take full opportunity when we engage with patients or have them engaged. And I think at the January meeting, Pierre, you talked a lot about the importance of getting the maximum information and benefit we can from every patient engagement. And so we all need to work together to help that, because it won't just happen. We need to use or develop the infrastructure to allow us to engage with those patients, to capture and collate the data, how we can involve researchers and academia in the process of evaluating it and reaching the appropriate conclusions, and how we can ultimately deliver that to regulators to deliver those drugs that patients are desperately needed. Critically in this process, the regulators have an important role, because often the data we're collecting is to support an application, and so for approval of a drug. And so it's really important. What we absolutely don't want to do is spend 3 years gathering data and then at the end found it wasn't the data that the regulator needed. We need the regulator involved at the beginning, in the middle, and at the end as a real partner in this, working with parties to identify what the data is and when we need it. Okay, if we could go to the next slide, please. Our collective responsibility. So I think this was just my last slide and it was a couple of questions. The first is just to ask whether we have the collective ambition to deliver new and appropriate pathways for ultra-rare diseases. And I choose the word ambition carefully, because we won't be stopped from achieving what we want to achieve by procedures. They don't stop you, nor do accepted practice or legislation or pharma or governments or time or money or lack of sleep. What stops us achieving what we want to achieve is lack of ambition. So do we have the ambition to deliver this? I think we do. I think the people in this room, from what I see and hear, have that ambition. And so if we have that ambition, then is it possible for us, and I'm already hearing lots of positive words about developing a real-time sort of working group structure that will allow us, all stakeholders, to take this forward and deliver what our patients really need. That's all I'm going to say. Thank you very much.
The EHE community has become an organization that has a strong research and development component, and I want to thank you for having funded it and shared it as that charity and driven it to this. It wasn't obvious to us, but with these elements, I think there is a structuring that has come out of these activities, and we need to come back as this idea of how such structuring can contribute to finding future solutions to that. So thank you very much. And I now hand over to Winan for introducing the next speakers. Thank you.
Yes, the next speaker is a well-esteemed colleague from the Royal Marsden in London. I would say he's a key opinion leader in the field of medical oncology and sarcoma. Robin Jones from the Royal Marsden, and he will discuss today identifying new drugs in ultra-rare indications and off-label use, which I think is an important topic here when it comes to ecosystems. So Robin, you're online. I can see you. The floor is yours.
Thank you, Vinan and Ralph, for the kind introduction, and also thanks to the EMA and EORTC and Sylvia for the invitation to give a brief presentation on identifying new drugs in ultra-rare indications and off-label use. My apologies for not being with you in person. British Airways cancelled my flight on the way to the airport today, so the earliest replacement flight they could offer me was 2:30 p.m. this afternoon. So I'm really sorry not to be with you in person. So next slide, please. So very briefly, I want to give my thoughts on identifying drugs in ultra-rare indications with a focus on sarcomas. And in terms of drug development and approval for ultra-rare sarcomas, in the context of my presentation title, there are 3 points to highlight, and I'm really pleased. Basically, I think everybody's on the same page that we need to change things, and we need to change our approach. And I think a lot of people will agree with what I have to say in this presentation. So the first point is that we need a better understanding of the biology of many of these ultra-rare cancers to enable rational drug development. But as Hugh and Harald have mentioned, this requires a lot of investment and research funding. And for decades, there's been a gross underfunding of both laboratory and clinical research in rare cancers, and this needs to change. The second point is that we clearly need more prospective clinical trials for rare cancers, but access to clinical trials is not equitable across countries and across sarcoma centres. In a way, I'm quite fortunate to work in a centre with an outstanding phase 1 trial unit, but despite this, many phase 1 trials now only recruit specific tumour types. And essentially now only a small proportion of my patients do participate in phase 1 trials compared to 10 or 15 years ago. And one of the questions is, how are we going to identify novel drugs or see a signal from a novel drug in sarcomas with limited enrollment in phase 1 trials? And the other point regarding clinical trials is the mismatch between the highly selected trial population and the composition of the general cancer population. And this is very relevant in the UK. And the other point in relation to sarcomas, and this has already been mentioned, is that it will be impossible to perform clinical trials for all 150 sarcoma subtypes. I mean, it's just not financially feasible. So again, something needs to change. And this leads to the main point of this presentation, that we need to focus on off-label drug use. For such a diverse group of rare cancers, but sarcomas and other rare cancers, off-label use of drugs with a sound biological rationale is essential in order to drive research and improve outcome. This is particularly relevant for sarcoma subtypes that we know have no standard conventional chemotherapy, or conventional chemotherapy clearly doesn't work. And as I mentioned, it's impossible to perform prospective trials for every single sarcoma subtype. So clearly more flexibility is required. And this interaction with the EMA and the FDA is so welcome in this regard, that you're open to discussion regarding alternative approaches. In addition, off-label drugs have a well-documented safety profile, and off-label drugs sometimes can inform the design of future prospective trials. So again, a major point for this presentation is that for rare cancers, we needed to develop an alternative approach, such as using real-world data to streamline drug approval. So next slide, please. So we need a better understanding of the biology of these diseases, as has been highlighted. And secondly, we need more clinical trials. But it has been highlighted, this requires a huge financial investment. And that's where the use of off-label drugs is so important in terms of informing the design of future studies and also hopefully aiding drug approval. Next slide, please. So an example in a rare cancer is the first case report of the use of imatinib in metastatic gastrointestinal stromal tumor. Prior to the publication of this case report, in many ways, GIST was a very rare diagnosis. There was a really good rationale for using imatinib off-label in metastatic GIST based on laboratory research published by Japanese researchers a few years previous to this publication. And this, of course, has led to subsequent prospective trials confirming the efficacy and safety of imatinib in this disease. And subsequent to this, it's become apparent that GIST is not as rare as it was previously thought, but is in fact one of the more common sarcoma subtypes. And this paradigm, this success story clearly won't be possible in all subtypes of sarcomas simply because of their profound rarity, but it highlights the importance of off-label drug use and what off-label drug use can lead to in the long run. Next slide, please. So in a way, this is not so much a provocative title to this slide. For rare cancers, we need a change of approach, and I think we're all in agreement regarding this. As I mentioned, for decades, there's been underinvestment in laboratory and clinical research in rare cancers, as highlighted in this editorial published in the British Journal of Cancer in 2017. And I don't really need to say more about this editorial apart from the title. In terms of rare cancers, the greatest inequality in cancer research and oncology treatments. So we really need to change things and improve outcomes, as Hugh and Pen have highlighted, for patients with rare cancers. And as they've mentioned, we need to do this quickly, and there needs to be a momentum. Next slide, please. So this is an interesting study published by a Dutch group recently. The study included over 170,000 patients with rare adult cancers, over 800,000 patients with common solid adult cancers. 18% of these were rare cancers. But the three major points I want to highlight here from this study is that the overall 5-year survival for rare cancers was 52%. The overall 5-year survival for common cancers was nearly 69%. The second point is for the two time periods from 1995 to 1999, compared to the time period from 2015 to 2019, the 5-year survival rate increase for rare cancers was from 46 to 52.6%, an increase of 6.4%, compared to 56.9% to 70% for common cancers, an increase of 13.2%. So almost a double in improvement for common cancers compared to rare cancers. And I sense that everybody is on the same page, that we need to change this approach, that we can't use an approach that works for common diseases, for common cancers, and just try and push that for rare cancers. We need to change the way that we do things in terms of drug development and drug approval. Again, this is not all due to drug development. There are issues with early diagnosis and, as I mentioned, understanding the biology of these diseases, but there's definitely scope for improvement in terms of the drug approval process. And the third point here is that for the majority of rare cancer entities, there was no improvement in 5-year survival in those two time points. Next slide, please. So this is not a representation of my discussion with Pierre or Francesco today. This is more a representation of my discussion with British Airways staff when they told me the flight was cancelled. So I think, essentially we're all on the same page and we all want the same thing in terms of improving outcomes for patients with rare cancers. Next slide, please. So in terms of sarcomas, representing about 1% of adult cancers, 150 different subtypes, as I mentioned, it's impossible. It's not financially feasible to conduct clinical trials for every single one of these subtypes, some of which are extremely rare. And just to be clear, I'm the biggest proponent of prospective clinical trials, having worked with David Cunningham, Johan de Bona, and Stan Kay. But it will be impossible to perform clinical trials for all of these very rare sarcoma subtypes. And given the disparity in outcomes between rare and common cancers, we clearly need to change. And this is where using drugs off-label comes into the equation. If there's a strong rationale for using off-label drugs, why deny patients access to novel agents, particularly as we will have a good idea of the safety profile of the drug or schedule being used for off-label use. So next slide, please. So just a few examples of off-label use and tying in with the biology of different sarcoma subtypes and the importance of using all three of those approaches that I mentioned in terms of a better understanding of the biology, clinical trials, and off-label drug use. This is a project led by Corey Painter in North America, where patients remotely shared clinical data and biospecimens for research. They studied angiosarcoma and identified a number of interesting potential therapeutic targets. But the one I want to focus on is the finding in angiosarcomas of the head and neck and scalp, where they identified that these tumors had a high tumor mutational burden and a dominant ultraviolet damage mutational signature. So consequently, a subset of angiosarcomas, ultraviolet damage may be a causative factor in development of the disease, and this leads to a potential role for checkpoint inhibitors. And 2 of the patients in this study were treated off-label with checkpoint inhibitors and had durable, exceptional responses to treatment. And this has led to subsequent publication of case series and clinical trials highlighting the benefit of checkpoint inhibitors in this subset of angiosarcomas. Next slide, please. And of course, Sylvia and the Italian sarcoma group have performed studies on the use of sirolimus off-label in epithelioid hemangioendothelioma, highlighting the efficacy of this agent in patients with multifocal and progressive disease with a median progression-free survival of 13 months and a median overall survival of 18.8 months. Again, highlighting the importance of off-label use of drugs in essentially a disease where no other agent really has activity, or none of the conventional agents that we use in sarcomas have activity. Next slide, please. So we clearly need a change of approach, and I think that the more frequent use of off-label use, where there's a clear rationale for using drugs off-label, where we've got a track record in other types of cancer, where the safety profile of the drug is known or well established, is important. As I mentioned, it's becoming very hard to enroll sarcoma patients onto phase 1 trials, and access to phase 1 trials is limited. As I mentioned, clinical trial entry should be encouraged, but this is clearly not possible for every patient with every rare sarcoma subtype. And if there's a good rationale for using drugs off-label, we should not deny patients the use of these drugs, and it can lead to subsequent clinical studies and even prospective clinical trials. And I think increasingly phase 1 trials enroll specific tumor types, and it's harder, in my opinion, for sarcoma patients to enter some of these trials. Next slide, please. So my 3 discussion points for everybody are that there's clearly an inequality of care between patients with rare and common cancers. This is clearly a complex problem, and there are a number of factors that play into this, including delayed diagnosis, the lack of research funding for laboratory research, but also drug development, and there's a need for more flexibility for rare cancers. And it's so good that we're having this discussion today with regulatory agencies. And thirdly, I think that off-label drug use is a critical approach in rare cancers, particularly if there is a clear rationale and good biological justification for off-label use. So with that, I'd say thank you, and I welcome your questions and comments.
Thank you, Robin, for this nice presentation. And I think what you highlight is that, yes, we need more flexibility, but also I remember Pan's words just, from an hour ago that, yes, we need to be more flexible, but we cannot lose quality of the data and lose the quality of, where we base approval of drugs for. So I think that will be a good discussion point for later. And to further go into this discussion, I would like to introduce Andrew Wagner, who is also key opinion leader in the field of sarcoma. He is from Boston, Dana-Farber, and he will discuss how we came to immunotherapy in a very rare type of sarcoma called low-grade fibromyxoid sarcomas. And then just to show how difficult it is to work with rare cancers, if this variant of sarcoma becomes more aggressive, more high-grade, we don't call it high-grade fibromyxoid sarcomas, but we call it sclerosing epithelioid fibrosarcoma. So a completely different name. Again, Andy, maybe you can explain why we did that, but maybe you can explain a little bit more how to deal with new drugs in rare cancers.
Thank you, Vinon. And I can take no credit or blame for the naming of these sarcomas. These are an unfortunate situation we all live in. I'd also like to share my thanks to the organization of this second workshop. It may seem a little strange that we have a couple of Americans here to visit the EMA, but I think it also represents how we feel that this is a super important topic to work on together. I think we have a shared response. Even though in the United States we have more access, for example, to off-label use of medications, there's a constant need to develop these drugs towards regulatory approval. I think it also shows that we all are highly committed to developing the best evidence possible to move from the land of anecdotes for drug therapy to the best evidence and the most feasible designs to lead to global access to these medications, not just in one country or another. So we're all, I think, highly committed to this. May I have the next slide, please? And these are just my disclosures. Go to the next slide. We chose to use this disease just as another example of how we're trying to move forward in developing better lines of evidence to lead to new treatments for these ultra-rare sarcomas. And I don't want to focus on this disease or a potential treatment approach in too much detail, but rather to use it as a case to illustrate both the challenges that we face in developing drugs, proving the efficacy or really establishing efficacy of drugs for these rare entities, but also to try to develop the models which we've heard from Hugh and from Robin already in terms of how to provide the best level of evidence for these diseases where randomization just will never be feasible. So just as way of introduction of these two related diseases, low-grade fibromyxoid sarcoma and sclerosing epithelioid fibrosarcoma, these are distinct diseases that are highly similar in their morphologic features, immunohistochemical staining, and molecular features as well. The low-grade fibromyxoid sarcoma is, as described in its name, a low-grade sarcoma. It tends to affect younger people. Both of these entities have high levels of expression of a protein called MUC4, and they have similar translocations of similar gene products, both highly consistent, not universal, but quite prevalent in these diseases. The more aggressive form of this disease, sclerosing epithelioid fibrosarcoma, tends to affect older patients and has a higher risk of metastasis, although even the low-grade fibromyxoid sarcoma can metastasize, often with a long disease-free period prior to that. But because of their real ultra rarity, it will never be feasible to perform a randomized study. And up until this year, there had been very limited data describing these entities in particular. 3 prior series of patients with really no more than 31 patients in any one of them. So what's happened through the global sarcoma community, and if you move to the next slide, please, are some retrospective series really collecting data from sarcoma centers of expertise throughout the world and describing the behavior of the tumors, both in terms of initial management as well as their risk for developing metastases, and the experience throughout the world of treatment with standard approaches to systemic treatment. So the French group published earlier this year from their sarcoma network 26 different centers. All their prevalent patients with these diseases that had been seen since 2010. So almost 15 years worth of experience. There's a total of 330 patients throughout the country in those 15 years. And of that, a limited number were a subset that actually developed metastases and were treated with systemic therapy. So a very small sample size, despite, again, 15 years of data collection. Among those treated, the anthracyclines appear to have the highest response rate. But the outcomes in general were still quite poor, with a median progression-free survival from metastases of about 18 months, with median overall survivals just in the number of a few years. The ultra-rare sarcoma working group out of the Connective Tissue Oncologic Society and the PUSH group similarly collected data from 28 centers throughout many of the continents. And this, going back to 2000, yielded almost 400 patients, 101 of whom had metastatic disease that met pretty prescribed entry criteria for data summation. They had to have been treated at a reference center. They had to have, the pathology had been reviewed at a reference center and had to have certain criteria to meet pathologic diagnosis. And so of these 101 patients who had metastatic disease, 32 had low-grade fibromyxoid sarcoma, 50 had sclerosing epithelioid fibrosarcoma, and 18 had a variant that I didn't describe in the introductory slide, which is a hybrid of having both components of it. So there's a third component, which is this disease that has both a low-grade and a high-grade component. Again, anthracyclines appear to have the longest progression-free survival in patients who had the hybrid disease, and pazopanib seemed to have the longest progression-free survival in the other 2 forms of the disease. But what was noted that there were 5 of the 9 patients with sclerosing epithelioid fibrosarcoma had prolonged stable disease when treated with immunotherapy agents. So that's the extent of our evidence for this disease. So standard drugs like anthracyclines, pazopanib, other agents, including gemcitabine, docetaxel, and trabectedin, were also summarized in these reports, have pretty limited benefit. And there's a very small signal of, it's hard to determine what it means, of patients with immunotherapy, treated with immunotherapy who had prolonged stable disease. So the question is, how can we investigate this further? How can we use this database of retrospective data collection of patients, an observation that immunotherapy may prolong disease stabilization in a subset of patients, and how do we turn that into enough evidence to make us all, from a clinical side, from a regulatory side, from a patient side, feel comfortable with this becoming a recommended therapy for management of this disease? Or how do we get to a point where we conclude that it is not an effective therapy and equally remove it from consideration and move away from anecdotes of individual practitioners deciding what types of medications they should use? So you go to the next slide, please. So I think we discussed this at the last meeting, but we, as a sarcoma community of experts, previously described a framework for defined criteria for retrospective series, describing the history of outcomes of patients with ultra-rare sarcomas. And this goes through a bunch of different steps, saying these are the levels of evidence that we should have to include these patients in the data collection. And I seem to have omitted the reference there, but you can see the Cancer Treatment Reviews publication on the left. So again, this uses, this gives us a framework that was used for that retrospective series I described, to give us some stricter criteria for what to include in the retrospective series, with all the caveats that come with it. It is still not standardized collection of data. It's not collected on a standardized time point. It's not collection of toxicity data. There's many caveats with retrospective data, as we all know. But the idea behind this is to help generate hypotheses. For example, maybe there's a role of immunotherapy in this disease. Okay, next slide, please. We ultimately want to get to that prospective interventional study using a very defined patient population. Again, using criteria to define make sure we're talking about the same disease, to make sure that there's common pathologic assessment, to make sure that the intervention is defined in terms of what we're studying. This ultimately will have a very small sample size. As I already described, it's taken 20 plus years to come up with 100 patients ever treated with this disorder. But also to define the assessments that will be in effect and the schedule of those assessments with prospectively collected endpoints. But some of the struggles, again, we talked about this last workshop as well, is if we see stable disease as the best outcome in a small single-arm study where randomization will never be feasible, how do we assess efficacy of the drug? And are there opportunities to use external control groups to help support the data that we're using? Now, we can turn back to the retrospective case series, but we would propose is something in between. I think Hugh alluded to this as well. Go to the next slide, please. Is to prospectively collect data in an observational approach. Now, observational could be untreated patients. It could be patients who are engaging with standard of care therapies outside of an interventional study, but where we can collect their data, defining again the patient population, have access to more patients than what we would accrue in a prospective interventional study, and to define the assessments really as standard of care assessments, but sort of the engagement of the community to say, let's do this type of standard of care assessments on this schedule and report it in a similar way with prospectively collected endpoints, again, being standard of care. Now, the variation will be here would be the interventions might be very different depending on where the patient is being treated. But the question is, can we use this to supplement the retrospective data in providing background information as to what to expect from this disease so that we can use that to help provide a control group, if you will, in a non-randomized prospective interventional study. And that's really the discussion we'd like to engage more in today and in the future. If you go to the next slide, a couple of questions that we can maybe discuss. One is, are prospectively collected data outside of a defined clinical intervention, would they be helpful in providing context for a comparative patient population, again, where randomization will never be possible? And what opportunities might we have in meetings with the EMA or FDA and other regulatory bodies to collaboratively develop feasible frameworks for the collection of these data from prospective observational studies? We don't want to leave it just to us as the academic community to design something if it won't be acceptable to the regulatory bodies. So what could we do to help strengthen the collection of these observational data, if they could be helpful, to help support the interpretation of single-arm interventional studies in these ultra-rare diseases? And that's, I think, the last slide. Thank you.
Sorry, thank you very much. And if before we go to the next speaker, if I can add to Andy, as a group, we have already started designing this prospective observational study. And this is going to be a study that involves from Taiwan to Canada, with everything in between. So for us is an urgent answer to understand what could be the best way to collect data to be used for development of new treatments. And what makes here everything even more difficult is that we do not have a patient community. For EHE, we have a global patient community that is really funding, supporting, helping us gathering the data and everything else, also attracting pharmaceutical company, while here we are, let's say, left alone. So patients are there, but they are not yet organized. So again, we are doing things, they are already ongoing. We really need urgent opportunity for meeting and understanding what could be done in the best way.
Yeah, because, and this is basically also for discussion, but I think this is a huge effort and it's a very good effort. But in the end, it doesn't make sense if it doesn't result into, you know, regulatory decision-making processes where we can actually give the drugs that we feel are working. So I think that's basically the summary of why we are here. And with that.
Yeah, very good. Andrew, thank you very much, Sylvia and Wien, and also to make the transition already to Pierre's presentation. Pierre, you introduced yourself already. You're a medical doctor trained and specialized in cardiology, deputy director at the French Medicines Agency for a Directorate, but you are the chair of the Oncology Working Party at the EMA and the vice chair of the Scientific Advice Working Party. So thank you for laying out, perhaps in just a few minutes before we enter into the discussion, what could be the development of medicines in ultra-rare cancers look like? Thank you, and over to you, Pierre.
Thanks, Ralph. And let's not be too much afraid. Yes, I'm a cardiologist, but I was a cardiologist many years ago. So after a very long collaboration with the oncologists, I became the chair of the Oncology Working Party. Forgive me. Anyone has a right to make a choice of the wrong orientations after all. No, but seriously speaking, I'm quite reassured by the previous speakers because I believe we are on the same line. There are points we must discuss to better specify the points of agreement and the points of progression. But I believe we are exactly on the same line. My presentation will be very short. Normally, it's the announcement for a very long presentation, but for once, it will be true. So the next one, please. I believe that this workshop, and we all agree with that, is not a collection of scientific advices. We are not here to say on that type of ultra-rare sarcoma, is my development acceptable or not, because it's much more complicated. But we will have examples, and the examples are always a nice opportunity to build something more general and to better understand where the most important points should be solved. We are not here to draft a guidance on the exception of the exception, because I'm responsible for the Oncology Working Party with many of the colleagues, some being in the room. We normally write a guideline for things that are common and for which the rules are obvious. And every time something is exceptional, it's not in the guideline, or it should not finish in the guideline. And this is why we have scientific advices after all. Because if a company or a group of physicians come to the Scientific Advisory Working Party and asking whether following the guideline line by line is a good idea, we cannot answer anything but yes. So useless. So people normally come and consult the SOAP to understand how they could deviate from guidelines. And sometimes it is for very good reasons. But we cannot write a guideline for every exceptional situation that would justify scientific advice. So we are not here for that. So what can we offer if it's not a guideline here, or if it's not a collection of scientific advice? We can offer something which is a general organization principles. I must say that I had the very nice opportunity of being selected as a coordinator for one repurposing advice, and Sylvia knows the adventure very well. It is on HE and Saramus adventure. And this was conceived at the start as being a almost normal scientific advice procedure. And we came with Oli Ten-Hun and the other coordinator, vice chair of the Oncology Working Party, as a matter of fact. We came to the conclusion very rapidly with the EMA colleagues that the scientific advice was not, as it is designed, the right procedure to be used. And we decided to almost destroy the procedure, inviting the colleagues to informal meetings to transform the so-called normal, very short periods of exchanges, very rigid, into something which is prolonged, which is an opportunity for adapting, changing our minds, and do things better, suitable for such an exceptional disease. And this was the start of a reflection on what's okay. Facing ultra-rare diseases is every time a different challenge with some points that are in common. But let's take the example Andy, you mentioned. What should I do when the best I could expect from a drug, which is apparently efficient, is stabilization of the disease? This could be a point for discussion. We could touch upon during our dialogue. But if we do not institute such a dialogue, we will never find an agreement. It would be a pity for everyone. So we have a toolbox at the European Medicines Agency. We have a toolbox with the ITF, Innovation Task Force. We have the scientific advices with the follow-up assistance, the follow-up, with the specific legislation for orphan diseases. We have plenty of prime designation as well. But those tools are in different boxes, sometimes not very easy to find. And a nice project could be to have a new box with those tools adapted to the needs available for anyone for an open dialogue. So I would say that, okay, a widely open toolbox could be a nice result of our interactions here. This is what we can offer, and it's not nothing. It could be very useful. What can physicians and patients offer? We already have that presented, experience, observations, science, real issues, many things for which we do not have very specific ideas here as regulators. And this amount of information with nice coordination is very important. So the next slide, I only selected on the following slide the most important words: flexibility, adaptability, dialogue, exchanges, toolbox, experience, observations, science, real issues. And all these things are between our hands collectively. And we have to bring each of us our gift under the Christmas tree to make things possible. Let's be clear, flexibility doesn't mean that we will lower our standards to something which will destroy the regulatory system. Flexibility is a flexibility of the approach. We must tailor our regulatory demands to the reality to such an extent that things are made possible but acceptable as well. But I believe that every disease, every condition, every development has its solution if needed, and we know how large the need is in some situations. The next slide, please. So we have opportunities, and I would say, what do regulators need? Because after all, I'm a regulator, even if I'm a physician. We must deal with the uncertainties around benefits and risk. If some of you had a look at the template for our assessment reports at the CHMP, the end is a summary around the uncertainties we can describe on the benefits and on the risks. And we come to positive conclusions provided that we can be convinced that these uncertainties are acceptable to such an extent that marketing authorization is the right solution. So retrospective observations are often, not always, are often not reliable to provide comprehensive evidence. This being said, they are not useless. To serve as a comparative group, not sure. I would prefer prospective as every regulator, but to give you an example, the natural history of a disease, and I'm not entering the discussion we will have after that, but it is very important. If we know that in that disease, to build up on your example, in that disease, we never observe stable disease. Stabilization is something that could be attributed to the drug effect only. I'm making a parallel here with our guideline that in some instances, when it is impossible to do anything else, we accept responses as a proof of a benefit because we know that we never observe spontaneous responses. And the response is the proof that the drug is doing something to the tumor. Is it enough? Not always, but if it's the best we can do, it's okay. If we can build a similar reasoning on never we observed any stabilization of the disease without a drug, and that drug stabilizes the disease in 50% of the patients, this must be explained by the influence of the drug on the disease. Why not? This is open for dialogue. It will never be in a guideline because it's much too exceptional, and we are not here for that. So I believe that for all these regulatory needs, we need collaboration between you and us, obviously, but even before. And here, I couldn't agree more than with the presentation performed by Hugh here. Patients are very important here. But I would say that the first part of the reasoning starts from the patients and from the physicians. But we need some organization of the collection of the data, prospective at the best. And this could be done through structures that we cannot find in the regulatory system. It is outside the regulatory system and before the submission. It could be during the consultation. Last point, I believe that an acceptable evidence could even start from off-label use, and especially, you may remember that at our first meeting, somebody came from the French agency to explain how we use the data in the compassionate use program in France to take that opportunity to collect prospective standardized evidence. If we could take the opportunity even of a compassionate use for standardizing the information we may get from the patients treated with a drug which has not demonstrated enough efficacy with reduced uncertainty to get marketing authorization, but enough to be used under a compassionate program, it could be a very nice opportunity to go further. So here, for once, the regulators could be important. It's not between the hands of the EMA because compassionate use is something which is national for the time being in Europe. I believe that in the States, it's much easier because in America, you have one agency responsible for everything, which simplifies the question. But I'm giving that as an example. But even at the EMA, we have the possibility of expressing an opinion on compassionate use, which has no direct obligatory consequences on the compassionate use in every country, but that could be a good starting point. So I'm only describing many solutions, many possibilities that could be used and that could be offered to the system. So my concluding words could be openness. What can the agency offer? Openness, flexibility, help. And this is something you can use as much as you can for the benefit of the patients, because it is what we all aim at. Every time you can provide us with acceptable plans, not even data before. And these plans must come from a common organization that will standardize, reduce heterogeneity as much as possible, collect reliable information, and then we will finish with a positive outcome, which is the dream of any one of us here. So thanks a lot for accepting that dialogue. I believe I'm doing nothing very new here by my speech, but only opening for the discussion we are having just before the lunch. So thanks a lot for that discussion, because we will learn, everyone will learn from each other, I think.
Thank you, Pierre, for this very interesting talk, and we will discuss that right now with all the speakers from the session, but also we have two more guests. Kit Roos, who is right here. Maybe you can briefly introduce yourself.
Yes, thanks. I'm Kit Roos for the role here, primarily chair of the EMA Methodology Working Party and a professor of biostatistics at the Radboud University Medical Center. So more than half of my time engaged also at the clinical research side. Actually, rare diseases in general is one of the particular attention areas.
Thank you, Kit. And we have online Martha Donoghue. Maybe you can introduce yourself as well. Martha.
Hi, good morning, good afternoon to you all. My name is Martha Donoghue. I'm a pediatric oncologist, and I'm the Associate Director for Pediatric Oncology and Rare Cancers in the Oncology Center of Excellence at the FDA.
Thank you, Martha. And to start with a citation of Robin Jones's presentation, rare cancers, we need a change of approach. I feel we all agree on that. I mean, when I listen to all the speakers, we all agree that we need a change of approach. So it's time for a different approach. And I would like to ask all the speakers to give a brief answer to this question. So what exactly do we need right now, or what do we need to change? If you have to mention one thing that is most urgent. Let's start with you.
I'm not sure it's changed, because I'm not sure it's actually happened very much. So I think we just need to get started, really seriously started. The one word for me is urgency.
Thank you, Robin. Urgency.
Yeah, I agree with Hugh and with Pan in terms of momentum, urgency, and get things done quickly. I think the thing that I find really, in a way, annoying about all of this is, for instance, sclerosing epithelioid fibrosarcoma. It would be so good if we could use the totality of the data, because there have been published case reports of actual dimensional responses to checkpoint inhibitors in this disease. But again, it's so difficult to bring all of that together, and I just wish we could be better in terms of the way that we collect real world evidence. So I think for me, the thing is the quality of the data that we as clinical researchers collect. And I know from the first meeting that we had, I really pushed in terms of what are the characteristics of, or what are the sort of defined criteria that we need for real world data to be acceptable by regulatory agencies. And it's such a difficult thing to pinpoint, but I just wish we could be better in terms of the quality of the data that we collect, but of course agree regarding the urgency as well.
So yeah, so basically you're saying we need to speak first about how to use existing data or use prospective databases, maybe even to use real world data.
And to be able to use the totality of the information that we have. So as I say, with regard to sclerosing epithelioid fibrosarcoma, there are a number of single institution series, clearly showing that conventional chemotherapy doesn't work. I mean, in our series from the Royal Marsden that a medical student published, that the median time to progression was, just about 2.5 months. So clearly, conventional chemotherapy doesn't work. And with all of these solitary case reports of responses to checkpoint inhibitors, as well as documented responses in prospective clinical trials, why can't we just use all of that or collect those data in a better way?
I just heard there's a toolbox here that we can open and we can see how flexible and adaptable we could be. Next speaker, or you want to comment? Andy.
I certainly agree that it'd be great to use those historical data that we have. I think one of the challenges that we face is maybe outside the regulatory environment, but it's how do we pull those data together? How do we, number one, record the data internally? We don't have a standardized format for doing that. And number two, how do we share it in a way? And as we're working on these initiatives for each different disease, we keep designing these platforms to do it, which are complex and legal and other challenges that come with it that just make it very difficult administratively to pull together the data. And Robin, as you highlighted, with over 100 different diagnoses, it's challenging to think that we could do this for every disease. But so if we could find a way to standardize the collection of data, even for our standard of care patients, and find a way to extract those data to a shared database, it would greatly facilitate the process. I know in the US, for example, the NCI is working on a process to try to do that, to try to pull data together from different centers into a common database. But there are challenges with that too. It's kind of just starting, but the idea is, can we better define certain key data elements so that we can extract them? Again, that's going forward, because we're not going to go back and do that all over again for our past patients, but it would be more helpful to have it in a more organized format to start with.
Thank you.
Yeah, I agree, Andy. It's really, as I say, I think the case of sclerosing epithelioid fibrosarcoma really illustrates this point well, where it's clear from these retrospective studies that conventional chemotherapy doesn't work, and there's definitely a signal that checkpoint inhibition is effective in this disease. But it would be so good if we could bring all of that together, all the information from prospective trials, as well as the individual case reports and case series that have been published. But as Andy says, it's really challenging.
Before we go to Kit and Martha, Sylvia.
Yeah. So as we said that we do not want to talk about a specific histology or disease, I think that what we can bring here as a community of physician and patient is the network and the collaboration, the willingness, and somehow also some infrastructure to collect the data. We are already working on it. For me, the comment and the question is that what Pierre said about, the toolbox wide open is really a great opportunity. And I would like to understand if this kind of open tool that we need to refine together could be the right opportunity to go more into detail about each type of data collection. So you ended by saying compassionate use data could be used. Okay. Outside a specific disease, can we meet and try to understand, possibly also with our friends from FDA, what kind of data collection is needed to have good compassionate use data to be used for, filing and approving a drug? Similarly, what kind of data can we collect in a prospective observational study on a very rare indication or tumor to make use of those data? So we, as a community for sarcoma, we refine some criteria for retrospective study. We would like to discuss with you those criteria. Are them enough or not? Are we collecting retrospective data in a good enough way or not? And this is a specific, it's like a RECIST, let's say. RECIST are applied to all tumors. That is the reason why maybe sometimes are not the best way to measure. So maybe also our retrospective data collection criteria can be improved. But we would like, as we are already collecting those data, specifically for all these exercises, if we are doing well or not, how can we improve, and how can we then make the best use of this data before entering the specific disease. So is this a tool, sorry, the name you used, I lose the slide, toolbox, the good opportunity, how can we organize such a kind of meeting and make the discussion more concrete in a way, even still we are, talking of very concrete things.
Maybe Kit can start with addressing this question. How can we, have a good quality enough database retrospectively, prospectively to make it acceptable in the toolbox? Or maybe Pierre.
If I may.
Yeah.
Because I believe I will introduce Kate's point, except if I'm confusing. I believe that speaking of real-world evidence could be a bit misleading because we are here with very exceptional patients suffering from exceptional diseases. We're collecting as much data as we can, retrospective or prospective, compassionate use or anything. Is it real-world evidence? Is it something which is already a clinical trial? It doesn't matter, indeed. I would make here a difference between what is retrospective and what is prospective. Not saying that retrospective is always unacceptable and prospective is magic solution. But let's come back to something which is the credo of the regulators. We must formulate hypotheses. Observing something and asking ourselves the question, do I believe in what I could see in the past, is not the best scientific approach. In the best case scenario, where you have anyone dying from a disease in 1 week, and you observe survivors retrospectively after 2 years, yes, you must believe it, but it's very exceptional. People knowing me as SOAP know that I say the best case scenario is not something that helps much. No, we must formulate hypotheses and check whether these hypotheses are true. This is the better scientific approach, and this is prospective. Real world or not, I do not mind. Use it in the context of a clinical trial, of a collection in a compassionate use context or anything like that. It doesn't matter, but I believe that prospectively testing the answers to our questions is much more reliable. But retrospective or anything like observations, not standardized, could be the best opportunity to generate these hypotheses and to test whether they are worth testing or not, and with which endpoints and which plans. I believe this is important to keep in mind.
I think Kit was eager to address this as well.
To some extent, I let's say the point you ask, which point would you pay attention to, was somewhat different, because I actually think that the data is yet but one factor in the uncertainty that we need to manage. So I very much start from the point that Pierre made on what we in the end do is assess benefit risk under a certain level of uncertainty. And with that level of uncertainty, we need to be comfortable, essentially. And I formulate this vaguely now. And my key point that I would, at least that I also feel as a challenge from a methodological point of view, we are quite good at articulating that uncertainty and even quantifying it. If it's a prospective randomized trial under type 1 error control, we have not articulated equally well if that situation cannot be achieved, and we deal with a prospective observational study with maybe a small clinical trial, or we have prospective observational data combined with a single-arm trial, which is essentially a different design. So one of the challenges I see is that I think we need to articulate that uncertainty for a less straightforward situation, which at the same time will allow us benefiting leveraging all available data. I think our classical thinking about uncertainty is also very much, we have one primary clinical endpoint. That's where you meet the bar or not. I think we're here in this setting, we need to put ourselves in a position that we are able to assess uncertainty, taking into account all available data, rather than one single endpoint. Actually, because I think I would love to stay prospective, I would love to stay confirmatory. I think in this setting, we can't fully achieve that. Any trial we will do will be partly confirmatory, but also partly to learn from, because it's diseases that we don't know that much of, and we should take that opportunity. So that's my take. There is methodological work to do here as well. That challenge I take. I would love to stay away from what's acceptable to regulators. I think we should have an honest discussion on which level of uncertainty do we think is acceptable from all perspectives. So both to make the decision, but also to make it available to patients and also to block other opportunities. Do we really think, given the uncertainty we have, that this is the best decision to take? So that's why I think those two come together for me. The methodology we need to develop to have a better assessment of data that's not so straightforward and needs a decision, and have an open conversation on the level of uncertainty, rather than is it yes or no acceptable.
And on this note, Kit, I think it would be very nice, since we have now heard the content questions, if we think about how we can structure and get it organized. There's the toolbox, there's what Sylvia, you said, the clinical researchers, the patients, the patient organizations, etc. So now I was wondering if I could offer the opportunity to Martha, if you want to, and then followed by Paolo Foggi online. Paolo, you are the chair of the Scientific Advice Working Party at the EMA, coming from the Italian Medicines Agency. And perhaps if you could think about what from your perspectives and experience, Martha, also a long time at the FDA now in oncology, how could we facilitate such discussions on the content? What is helpful to get to a discussion that informs on the quality and on the options and how to use that? But please take it more broadly as an invitation to come in and suggest from your perspective what is helpful to structure the discussion and move forward. Thank you.
Thank you so much for the question. It's a very big question. I think that there are multiple ways for us to have ongoing conversations, communications, and dialogue, both looking at the big picture areas that need to be addressed to benefit drug development for ultra-rare cancers, including ultra-rare sarcomas as a whole, sort of trying to figure out areas of agreement and approaches that are acceptable, to multiple regulatory agencies and communities to think about and implement when addressing how to effectively develop new treatments for very rare cancers. And I think those types of work efforts include, really probably sequential, detailed, focused discussions on common data elements, data sharing. What are the overarching principles that we think are really important to keep in mind when thinking about using real-world evidence or data from compassionate use trials to support drug development? So I think, we have existing channels and ways that we can work together in that way. And then there are sort of the more specific discussions that one needs to have when thinking about a specific drug development program, which needs to be highly contextual and thinking about which of the tools, which of the approaches are most appropriate in a given disease context, and thinking about the biology of the cancer, the unmet medical need, the scientific information we have supporting the potential mechanism of action of a particular therapy. And those types of discussions, we also have ways to coordinate and work together toward as well. But they do differ. And, I think these are often highly complex discussions that require a lot of thinking and more than one interaction often to reach agreement. But I think the important concept for me for both is that we think prospectively and ahead of time on this, as opposed to gathering data and trying to repackage it, which sometimes works to help support a marketing authorization, but also can be fraught with risk and can ultimately waste time. So I think, trying to make sure that we have avenues available that are clear to everyone and for communication as early as possible and as often, I think are vital. We do have, many examples of successful drug development programs for pretty rare cancers, but oftentimes they're kind of one-off approaches, right? Sometimes, very unique to a situation, and sometimes they've been successful. And other times we've seen examples where we've had like long, lengthy development programs that haven't resulted in anything, and some of that's due to lost opportunity over time. So that's a long-winded, probably circuitous answer to your question, but I do think that we have existing mechanisms in place. It's just a matter of how to leverage them and kind of have this ongoing productive conversation to make sure we're fully leveraging the opportunities we have.
Thank you very much, Marta. I think words that stuck with me were sequential, detailed discussions, non-clinical unmet needs, mechanism of action. And I think this points to the need for a sort of a project plan, or when we thought of it, it's sort of an evidence generation plan that puts these elements together, that facilitates us discussion. So really looking at it from the various angles. And you also mentioned there are examples that have been successful. Maybe they can serve also as instructive examples how to structure such an evidence generation plan, thinking there are several options, several avenues, as you said, that may need to be explored, and then after exploration, one can see if it works to merge data or not. Paolo, please, over to you. And I already introduced you. Please, if you could also maybe expand on this matter. What helps the Scientific Advice Working Party discussing cases? Thank you.
Oh, thanks, Ralph, and thanks all. And really interesting discussion. And of course, I guess that we all agree that with the first presentation, the second presentation was about starting from off-label use to detect signals, and then of course that would be then there should be a way to move in from anecdotal evidence to the highest quality evidence possible that it is. That should be that my main message would be, and I guess it's very clear to the researchers in the field, that no single data should be lost. Because then I guess that Andrew had the very important question, how to assess single-arm study if no objective shrinkage of tumors is there. And then it comes to the fact that we need to do some sort of contextualization. So then we can speak a lot about how can we use external control group or things like that. But this is the key issue, because we have a lot of examples in which even in rare diseases, rare tumors, single-arm trials were enough, and that's uncontextual, and it's clearly easy when you have a high over response rate. But what if you don't see that? So the key is the data, possibly the collection of prospective data, and possibly the collection of data with an accuracy and a quality that could in the end match the regulatory expectation. And this is, to me, something in which we could collaborate in general on the wide, on the broad, regardless the single type of sarcomas, because contextualize and it's not easy for a heterogeneous group of tumors such as sarcomas as. And I guess that we have guidelines in place to guide how registries or collection of real-world data should be in terms of data we want to see, and also in terms of data quality. And I guess that we also have procedure in place to drive the development in this regard. So to me, on top of what has been said, I would say that a dialogue to align on what would be the minimum requirements of the data, how we could think that all the data that we collect could be used for any specific context of use, being this an external control or to contextualize. Or so to me, it's something that should be part of the discussion in the long run. But there's I guess I stop here.
Well, thank you so much. And what sticks with me is your phrase, no data should be lost. And I like that phrase. You know, there's so much data out there, but it should not be lost and it should be taken into account. The question is how, and maybe I would like to invite Denis Lacombe as well to comment, because I know EORTC, director of EORTC, and I know rare cancers is a key focus point of EORTC, and a new program will be developed to collect prospective data. Denis, what is your vision on this, and how can play EORTC a role there? And also, which stakeholders do we still miss? For example, pharma is not right here on the table, maybe others as well, the EU. So maybe you can comment on that.
Yes, and thank you for these discussions. So for those of you who don't know me, I'm Denis Lacombe. I'm the CEO of the EORTC. First of all, I would like to say that I agree that we have to re-engineer everything because, of course, we are facing different challenges. And for me, my learnings of this morning is that I've tried to structure a little bit what we have to re-engineer. And I think as academics, sometimes we don't understand what is the needs of the downstream stakeholders that we have to deal with. And I have tried during the discussion to a bit structure that. I think we have to re-engineer at the level of the organization and the data we are going to collect. And I think there is something that is not for negotiation today. It's clear from the regulator. We have to organize prospectively data collection, forget about retrospective data, otherwise we don't meet the regulator at the end of the day. The second thing is what Keith has told us is about methodology research. I think we have indeed to address that, because at the end of the day, indeed, there is a regulator, and what you just said, Winan, I think we will need, of course, at some point in time, the pharma sector, if we want to make. And if also we want this data to be regulatory acceptable, but also if we want the pharma sector to embark with us, this data have to be of regulatory quality, and that you don't organize from scratch on a new program. I think you have to build on infrastructures, on solutions who have already this, I would say, this culture, this capacity in place. Otherwise, you will not address what you told us, Hugh, is the urgency, because you will have to create everything from scratch, and that I would really not advise. Having said that, and we have some example at EORTC, I don't think it's infeasible to create agnostic infrastructure that absorb some of these challenges. And then we can see, of course, how they can be customized for drugs or for some specific situation. So that would be my first reaction. We have to see the entirety of the challenge. And again, these three different levels, again, organization of data, methodology research, regulatory acceptability, and suitability for the pharma partner. And if we don't see all that at the beginning, I think we are going to not to deliver what is needed. So that's my first analysis from the discussion of this morning. And again, there are solutions. I think there are a lot of things existing, and we can work from there. Thank you.
Sylvia.
Yeah. Thank you, Denise. I have now maybe a question for Pierre, Keith, Francesco. And the question is, how do you see the next step for this kind of workshop? You see more that we take a disease as an example, like the one presented by Andy. So we have this retrospective data, some preliminary ideas that something could work. We would like to, as we did for EHE sirolimus, to confirm in a prospective way. We could present a proposal for an observational data collection, prospective data collection. And then we discuss this more as an example than, without the pharmaceutical company already here in the room, because I think that the perception of the risk in investing money on a study on any drug in such an indication is still too big because of the uncertainty of how the results will be interpreted and used from the regulatory point of view. So I think that we still need to work out a little bit to agree a little bit on the methodology and the quality of the data that we want to collect. Or you see more a workshop in which we talk, for example, more in detail of what Kit was saying. So what kind of different methodology? Because in my view, if we have a concrete example with some data, even though limited, is easier from that example to run a discussion. For sure, that example will not cover all ultra-rare sarcoma, even less all ultra-rare cancers, but is a starting point, maybe, discussing an aggressive sarcoma, very rare, blah, 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 blah. So what could be the next step to make a concrete use of the open toolbox that Pierre was mentioning, and as a group we really do not want at all to miss?
Well, Pierre or Francesco or Kit, I don't know.
So I'm a regulator, but I'm not in the governance of the EMA, so I'm relatively free in my answers, even being unrealistic. If you want to build on an example, I just mentioned the interaction we had on EHE and on the repurposing. I believe that this is the right model we should build on, because what have we performed so far? A very adaptive consultation between a group of academics, the patients, and a bunch of regulators trying to find the best solution on a continuous dialogue and possibly a plan for development that will bring at the best a package which could be acceptable to the CHMP for a positive discussion. Is this the right model that would cover for everything? I have to discuss with my colleagues from the EMA, with Paolo, with colleagues from the Scientific Advisory Party, from many other colleagues to decide whether this is enough or whether the entry into such a program should be better, the ITF or any other structure. However, I believe that as soon as we identify a proposal for development in any ultra-rare disease, we must, and this is our responsibility, we must adapt. Whichever the entry point, we must adapt our responses to the needs of this specific singular development without adhering to anything too formal. This being said, it's very easy. It's very easy. And not to reveal something which is confidential, I don't think so. We are, and I'm speaking here under Paolo's control, we're considering adapting processes such as discussion meetings, the times for the discussion in the SOAP, to the specific needs of the developments we observe, not necessarily having after 1 month a discussion meeting for 90 minutes and so on. No, this doesn't work necessarily. We sometimes need more time. We sometimes need to meet earlier. Maybe a shorter interaction or a whole afternoon could be okay. So you see what I mean. I believe that whichever the entry, it is up to us to bring the tools taken from the right box to people who come and do not know what we can propose. So the entry could be the repurposing process. It could be any other repurposing process. Maybe it could be a good idea that as soon as somebody at the EMA identifies such a proposal, we build a team that will make people available for the discussion, destroying possibly the rigid structures, or I would say not destroying, but adapting them very, very, very with much flexibility to the needs. This would be the proposal. The example, we already have one. Getting a second one would be even better, because we could consider where the differences are and how we can adapt to the differences. Let's not build something which is a new, inflexible and very rigid structure based on one example only. This would be my answer. You will understand that for that, we will have to have internal discussions to make things possible and to involve in many other structures. The SOAP is an example, the Oncology Working Party is another example, the Methodology Working Party is a third example, the COMP is another example. Many people could be involved in the discussion. Let's not avoid any discussion just because it's not described in the system. This would be my proposal. This would be my proposal. And I believe it's relatively easy. No need to build a new legislation or anything like that for this. It is between people of a goodwill, I would say. So yes, I believe it is right possible, and that toolbox, it's up to us to give it to you.
Paolo, your hand is raised.
Now, yes, I wanted to complement a little bit what Pierre said. I think that, yeah, starting from concrete examples is the best way forward. And then I'm not now practically, I think that we should reason a little bit internally and perhaps with the EMA colleagues, of course. But yeah, starting. But I wanted to say this. On the one hand, I guess we could start from products. On the other hand, I would like to start also a dialogue on the data collection platform that are over there, because to me, one thing is reasoning on a concrete example of a product and to understand how this could fit and how, starting from what we have, it could then go to a marketing authorization. What do we need? But looking forward and prospectively, I guess that the key changer here is how we can use the data that we can collect in the real life, and this is something that would be really the key that might change the future. And then on this, I guess that we should reason, what is the context of use? What are they fit for? What are the way you are collecting this data? And does this fit with the regulatory requirements? Can we get an alignment of what the expectation the regulatory ours and what the people on the field is doing? And to me, this is broader and this in parallel to looking at single examples. To me, this is a discussion that we should have. And of course, qualification is the normal procedure that we have for this kind of stuff. But I'm not saying that we should go there, but yeah, a dialogue in this direction should be run in parallel with specific examples and should be very concrete. And I stop here.
Yeah.
And just this is maybe not in the field of the EMA properly, but let's start a discussion. I remember that last time we met in January, we had a presentation of the compassionate use, and somebody from the European Commission was very interested in that model. And maybe another initiative which would be possible would be to discuss with the Commission to have the EMA in the driving seat from the scientific point of view, and the European Commission encouraging the member states to start with a compassionate use that would be recommended and operated in any country that can afford the financial efforts of the compassionate use. But in order to make it possible that patients included in compassionate use in Ireland, in Cyprus, in Denmark, follow a very similar pathway and are an opportunity to collect very similar data is this real world evidence? Is this prospective clinical trial? Is it a compassionate use? I do not care. But it could be something we could open, maybe a discussion with the European Commission on that, and to find a person who was interested in that, because I think it would be an opportunity indeed.
I completely agree, and I know Denis is also, you know, very much promoting this, and it's also commented in the chat that there's a huge difference between the countries when it comes to compassionate use, which is really a problem, I think. And speaking to the Commission to align that will be a huge effort, and it will be an accomplishment. We need to wrap up. Sylvia, Kit, and then I will close.
So for me, I was giving to Kit.
I will be brief, also given where we are in terms of the agenda. I would like to add, in engaging in the discussion, I say from a methodological perspective, it also makes a lot of sense to do that by example and have a very thorough assessment of examples. And I'm not, let's say, not too deeply worried about the exact structure in which we do it. I do think it's important also on the regulatory side, with which mindset do we enter those discussions? Because if you stay close to the standards, it will be actually guiding applicants and discussants to within the current regulatory thinking. And I think I would be open to actually also enter it with a mindset what needs to change or be added on our end to actually meet the needs that are there for these very ultra rare cancers. So I think partly it's structure, partly it's also very much how do we frame our discussions that we will have on particular use cases, because we are trying to push the envelope here and there, and that I think actually that needs to happen on both sides.
Yeah, the question is how to proceed. Before I close up, Harold, your hand is raised too. Comment on this?
Yeah, thank you. A very short one. Easy to agree with almost everything that has been said here. And Pierre has laid out that we have quite a number of different groups, some well established, some still to be collected, to be gathered, to tackle that and to push that ahead. One thing I'm afraid was missing, and let me introduce that. In the end, there is no way to get around a positive opinion as the watershed. And here it will be important that early on, and I missed that, you get a buy-in from CHMP and CHMP members. Early on, because there are one thing, looking at the data that are presented. And the other thing we always have in our discussions is that we need to be consistent. It is a matter of fairness to see what has been done before and not deviate from that unless we have very good, transparent and justifiable reasons. And if we fail to get this buy-in early on, there may be a situation where we say, oh, well, that's all nice and beautiful, and we would like to do that, but that has not been a deliberate announced and made publicly knowledge of a change in our criteria. Therefore, committee members may feel bound to previous decisions. Yeah. So please do not misunderstand that as putting up a hurdle. What I'm trying to say is be aware that that will be early on a good thing to include, to inform, and to get the buy-in, and not do that only when the thing is ready, because then this consistency issue, and now that has not been announced publicly, so we cannot change, might get in the way. Please, just as an additional point, keep that in mind, creates a linkage. We have that. We have these tools for that. Paolo is each and every month at CHMP presenting the most interesting or even controversial issues from scientific advice. Yeah, that might be one of the channels that can be used, but please build that in early on to avoid a nasty surprise later on. Thank you.
Pierre?
No, I couldn't agree more, Harold. So we have two systems, one which does exist already, offering systematically a discussion at the CHMP for any development on which we are taking decisions. So it will be in the frame of the presentation we have at each CHMP of the scientific advice procedures. It could be the first example. Another proposal would be to take an example of the prime where we have somebody from the CHMP involved at the starting of the procedure, and to be the person in charge of checking the consistency and the compatibility of the development with the CHMP requirements. One of these two systems or both systems. Let's be creative, but we already have such systems in place. So we are not creating anything new. But yes, you're right. The CHMP must be on board as early as possible, because otherwise we would miss our goal, and it would be a pity.
So I think first lesson from myself is interactions with regulators will be in workshops on general topics, but also on cases, as we said. EMA especially supports not-for-profit developers, and that is a term we use for clinical researchers, perhaps like yourself. But it takes more than regulators interacting with clinical researchers. And I want to bring in brief points from the experience. It's important for clinical researchers to take out professional services. There is often local support in universities for both technology transfer or for regulatory support even. That depends on the faculty, but it is growing, and we see this really as a change in the landscape. There is so much local support that should be leveraged. In particular, as a first thing to come up perhaps with a strategy or a regulatory plan. So what are these elements? Where are you? Can one consult locally already with people who know about the structured thinking, how it goes? Second, it's also important to build in methodology expertise into the project. And I think that is what we heard already, but it's not just a consultation on the power and the p-values. It's a consultation that is integral, that is about the methods, and that matures the methodological thinking so that when we have the regulatory interactions, we can build on that. We can build on already the integrated methodological advice, having looked into challenges and options, etc. And that should lead also to come up with an evidence generation plan, so that we are not discussing a study, but the purpose, the background, etc. And that really helps to give it perspective and anticipate that critical thinking that you heard mentioning about. So I will stop here, but the last point, the evidence generation plan is important, because that can then be a vehicle for facilitating the discussion. And we will help you asking the right questions from the regulators. Sounds circular, but yes, I think that is what is our experience in the past. When we see these plans, we can help spot the difficult points that we would like to make sure to discuss well with you. Thank you.
Well, to wrap up before lunch, Sylvia, you want to? Oh,
I think we have to go for lunch.
Yeah, exactly. So yeah, because everyone is hungry. So I think there's a lot of enthusiasm here in the room, and I think we basically all think together, and that we heard there's a lot of tools that we can use already. But I also want to remember the words of you. There's an urgency, right? Because if we, every year or every, month even that we wait any longer, patients might die. And I think we should think about over lunch and in the afternoon how to proceed. When to proceed. What kind of meetings would we have, but also who is going to collect the data. For example, EORTC might be very well positioned to collect the prospective data, but it needs to be generated, needs to be funded. I mean, lots of issues to think about and to discuss. And last but not least, I think we need to think about harmonization of everything, the data between FDA, EMA, US, and Europe, but within the EU countries, between the different diseases and different rare cancers. So with that, think it over lunch. Thank you for this discussion, and we will reconvene at 2.
Okay, so let's restart. And I'm very pleased to moderate the second session of today's workshop with Denis Lacombe. So my name is Caroline Volz, and I work in the hematology and ATMP office. So today we are going to look at practical examples, and this is also in the line of the last workshop we have had, and especially to understand what we have learned so far. And I think it's always important to discuss specific cases so that it gets very concrete on the next steps and to come with possible action. And the first presentation is looking at endpoints and what is important for patients in addition to the RECIST criteria and overall survival. And I'm very pleased to introduce Professor Gerard van Ortmersen, I hope I pronounce it right, who is the co-chair of the Sarcoma Patients Global Network. So Gerard, over to you. Thank you.
Thank you very much. Yes, my name card says Leiden University, but the affiliation that is really important to this meeting is that I'm the co-chair of SPAN. So SPAN is the global network of patient groups of sarcoma patients all over the world, mostly national organizations, but there are some also are specific for specific subtypes. And in countries where there is not an organization, patients can also become an affiliated member of SPAN. We have now more than 60 member groups from all parts of the world. Next, please. We recently we performed a survey among the members, and the results we organized that together with Binet van der Graaf and her group, and the main results were presented at last year's CTOS in Dublin. And I just want to mention that it's clear that there are many unmet needs with sarcoma patients, and that has been emphasized already a lot today. But what I would like to specifically mention is that, as can be expected, one of the questions, the wishes of the patients is that more research is being done, and in particular on specific subgroups. But also another point is that there should be more emphasis on quality of life. And I think that is in particular of importance for the discussion we have here. Next, please. And if we look at the topic of this workshop, it's clear that there is a need for new treatments and new methods, baskets, repurposing, off-label application of drugs. But also we need other criteria in addition to RECIST and overall survival. And quality of life is a very important topic for patients. Next, please. In this respect, patient-reported outcomes are very important, I think. In addition to the clinical data. And then we talk about side effects, adverse effects, symptomatic side effects, but also overall side effect impact and physical function and social function. These are all important for patients, for their quality of life and how they can still function as long as they live with the disease. Next, please. There may be hidden side effects, and some adverse effects are challenging to observe, like fatigue. And therefore, if we do not have the patient perspective, that may lead to underestimation of the bad effect of side effects. In particular, if we do not observe things like fatigue or more difficult to see side effects, that will hinder the development of supportive measures. Next, please. And for this reason, patient-reported outcomes are very important. We live also in the era of digital revolution, and There are methods being developed right now for getting the patient-reported outcomes, but we need to further develop those. And in the future, smartphones may be useful, and also the internet for using for patient-reported outcomes, and it opens new opportunities for longitudinal data collection. Certainly, internet and the new developments that we see and which we see also are accelerated in the development right now as artificial intelligence. They become available to everybody. And there are certainly dangers in using these new tools, but for sure it also empowers patients. And we have seen already examples, I think, this morning also, for instance, in Hugh Leonard's presentation on EHE. Next, please. Patients take action together, and this is very much facilitated by the internet. With the very rare subtypes, people are lost if they get a diagnosis. They have never heard of the disease. They don't know anybody else with the disease. So often they go on the internet and they are looking for the disease, and they find peers in other countries, and they connect, and they start to collaborate also. We see discussion forums, and for many very rare subtypes, you can find, for instance, Facebook groups of patients that communicate, share their experience, and share all the data on their treatment, but also their daily life. We have many examples. EHE mentioned already, we heard about this morning, but also chordoma is such an example, and for GIST as well. A couple of years ago, I became active myself on the GIST Facebook group, and I found that there is really a goldmine of information shared on such a platform. And I started to collaborate with researchers at Leiden University in advanced computer science and data science and natural language processing, and also with Hans Gelderblom of Leiden University Medical Center. Next, please. And we started to look whether we could make any sense of the huge amount of discussions on this Facebook group. There are more than 6,000 members and a lot of posts. Next, please. And we did a PhD project where we analyzed the side effects that people were talking about. So this could be seen as patient-reported outcomes. This is an example of a result that we were able to get for avapritinib, for one of the subtypes of GIST. Next, please. We compared the results that we obtained with the results of the trial. And here you can see that for certain adverse events in the trial, the effect of the side effect is underestimated. For instance, impaired cognition, which means memory loss. It was graded at 8 in the trial, whereas if we look at the patient discussions, it is the worst side effect that they experience, just as an example. Next, please. We also compared the results, also to validate the results, with again with some researchers from the group of Wienet van der Graaf that used a survey with EORTC questionnaire, and we found a good comparison with this. This just is an example of what we can do using patient discussions, and this might in the future be a source for also getting patient-reported outcomes. Next, please. My conclusions, I made an embarrassing mistake when I wrote this late at night. It's important to look at patient-reported outcomes is the main conclusion, of course, but the last one there I made a mistake. Collaboration between clinicians, researchers. Well, that is evident that we have that, but I meant here the collaboration with patient groups, and I echo Hugh's comment this morning. The patient community is a powerful partner. Thank you.
Thank you for emphasizing indeed the importance of quality of life and patient-reported outcome. I think it came already also this morning in the discussion. It's my pleasure now to introduce William Tapp, our second US speaker. So thank you to both of you actually for making it all the way. And indeed, I think we need examples because we need to see how we are going to picture that into the learnings of this morning and how we bring that together in the discussion later on. So you are going to speak about the use of real-world data to complement clinical study and more specifically about alveolar soft part sarcoma. Yes. Thank you.
So thank you very much. Again, I'm really excited to be here and just that we have this collaborative thinking about rare cancers. I think, what I've been able to take home from this over the past, several meetings is that we all agree that, this is a huge problem. We all agree that this is a huge opportunity. We all agree that we bring different expertise to the table. And I'm very excited to hear how everyone is going back and really working on, their areas of expertise. Pierre, just hearing how you're going back and thinking about processes and saying, what can we bring to the table? And in part, this presentation is a little bit about that as well, because I think we've heard very clearly the need in ultra-rare cancers to think about not only counterfactual data, but how can we come up with prospective data to try to really complement regulatory packages or our understanding of drugs. So I just want to talk a little bit about how we've been doing that in our community and why, and talk of a few examples. So most of the investigators here in the room, and so maybe we can do next slide, have been really working on sarcomas and ultra-rare cancers for about 25 years. And I think all of us have grown up in this process. And when we first started, it was the exception to have a large international study. And large in sarcoma, maybe 99 sites worldwide, but trying to accrue, 500 to 750 patients. And the group really here, within the last 15 years, started to bring these large clinical trials to fruition. And there's just about 5 agents and 5 trials that we ran, and probably about greater than 2,000 patients. But unfortunately, they were all negative trials, right? So it was a big setback for us, but we spent a lot of time to understand why they were negative trials. And there's some obvious things that came out of it. One is that in some of these large trials where we're accruing 750 or 800 patients, we were including over 80 different or 40 different diseases within those trials. So, if you think about doing that in sarcoma, it's just like running a pan-cancer study. And it's not just something that we would expect to see positive results. The other thing, when we talk about using sometimes 100 sites worldwide to accrue these trials, you have to think about variations in practice patterns. And many of these trials had overall survival endpoints. And you can just think about all of the different agents that are available in diseases and that variation between countries. And so in a composite endpoint like overall survival, that's going to greatly affect it there. But if you go to the next slide. I think one of the striking things that came out of it for me was to understand why these trials were negative when they were often predicated on some very strong phase 1 and some phase 2 data. And what we became to realize is that we're often not studying the same patient populations in earlier, smaller, well-controlled trials where we can really select patients and scrutinize them and understand why they're responding. But when we go to bigger and larger studies, unfortunately, this gets diluted out and we often lose signals and consequently lose efficacious drugs. So next slide. So while we were coming to this realization, it started also to bring into the genetic and molecular era of what we would do. And there are multiple efforts, and this is just one of them, where we began to look broadly across all of the sarcoma subtypes. And we really began to understand how different these diseases were on a genetic level. And what was also a little humbling was some of these subtypes where we may only see a few hundred patients each year, they're striking genetic heterogeneity within the disease itself. So we're faced with a lot of variables that we have to include, especially when we think about developing targeted therapies in these diseases. Next slide. So right around the same time, you've heard that the community was getting together and trying to think about how to do this in ultra-rare sarcomas. So when the ultra-rare sarcoma working group was put together, the first goal of it was even just to define ultra-rare cancers, right? And we were defining them as an incidence of less than 1 per million, right? So that's, again, 200, 300 patients diagnosed in the United States each year. But that's over 70 different cancers that we're now faced to understanding how to develop research programs for and treatment strategies for our patients. Next slide. And this is really challenging because the truth is that many of these diseases that we're trying to study are actually newly defined entities. You know, with all of these new molecular and genetic techniques, our pathologists are finding new diseases all of the time that as experts, we're expected to understand how to treat. And patients with these diseases, they come to our centers not just to say we've heard of it, but that we know how to treat it. And inherent to that is we don't truly understand the natural history of the disease. We don't understand what patients go through. We don't understand how the disease affects them. And then when you think about that, how can we develop drugs for this disease if we don't truly understand what we need to do to help a patient? And as I mentioned before, many of these diseases have a reasonable amount of genomic and clinical variability, and all of these need to be encompassed when we think about drug development strategies. And then next slide. And I thought this was an interesting publication that just came out in the JCO by Mark Fleury's group, actually looking at the percentage of cancer patients who actually enroll in therapeutic clinical trials. It's just about 7%. So if we think about only a few hundred people with a disease being diagnosed each year, and we talk about 7% enrollment in clinical trials, are we going to have the numbers to run the types of studies that we need to run, right? So this is one of the issues. But we've had a lot of successes as we started to move away from these larger clinical trials. So next slide. One of the successes was in a disease called epithelioid sarcoma, which again is an ultra-rare sarcoma, probably about 200 people diagnosed in the United States each year. Like many of these diseases, it affects a young patient population. And we haven't spoken about that today, but we talk about ultra-rare cancers. We're talking often about the teens, the 20s, and the 30s, right? So this is a really important patient population that we have to affect. But there are different clinical variants to this disease. There's a disease that's known as a distal epithelioid sarcoma, where patients often present with painful lesions in their distal extremities. They can actually ulcer, and then they can spread intransiently. They recur, and they can spread via the lymph node. And then there's a proximal type, which tends to happen in more of the proximal extremities, the girdle. And these can be exceedingly aggressive diseases that can metastasize and patients die of them. Interestingly, they all have very similar genomics. They have loss of what's known as INI1 or SMARCB1. This is a big epigenetic modifier, and it preferentially has these tumors relying on the PRC2 and the EZH2 complex. And so next slide. Why was this important? Because companies were developing EZH2 inhibitors. So it gave us an opportunity, like with many of the diseases we study, due to the genetic variability, to find drugs specific for the disease. And so there was a phase 1 trial, and then seeing some responses in epithelioid sarcoma, we were able to get, and we collectively in the community were able to get an expansion cohort where we enrolled about 35 patients. And we actually saw some reasonable responses, an overall response rate in 15% with duration of response of about 14 months. Now, this is a disease where we typically don't see responses in chemotherapy. So this actually led to accelerated approval of the drug after an ODAC meeting in the FDA. So this drug is now available in the United States, and it's used all the time, and we're thankful for that. But in this data set, we cannot actually determine how we should use this drug. So next slide. The big question is, how do we use tazemetostat? And so it's being used in the front line, it's being used in the subsequent line of therapy, it's being used in combination with agents. We don't know if preferentially we should be using it in the proximal or the distal subtype, right? So we need to answer these questions, but we have the drug to do that. Next slide. A confirmatory study was being run, but it was in a totally different patient population. So we don't know even if this is going to accrue, so we cannot necessarily look for the confirmatory study to answer these questions for us. This is something we need to do as the community. Next slide. Another disease that I just want to mention briefly is alveolar soft part sarcoma. So this too is another very dangerous sarcoma that affects younger patients. There's a very characteristic genetic translocation in this disease, but despite the same genetic translocation, we have different clinical presentations. There is a type that develops in the head and neck, and when patients present with that, they often present with hundreds of nodules within their lung. Interestingly, it spares the apices of the lung and is mostly in the lower parts of the lung. Chemorefractory disease. There's also another clinical variant of this that actually affects the, again, more axial skeleton. And that disease can be very aggressive, very few nodules in the lung. We can see spread there, but it often spreads to the liver. So this is a disease that we've had nothing to treat for the longest time. And all of a sudden, we started to hear anecdotal reports of responses to immunotherapy. A priori, we would never have thought this because it was such a genetically simple disease. Next slide. So we started to put patients on clinical trials with immunotherapy. This is just a handful of patients that were put on a CTLA-4 and a PD-1 study. And in the 10 patients with alveolar soft parts, you actually saw a 40% response rate. Based on these data, CTLA-4 and PD-1 are on the NCCN guidelines in the United States, so we can use these drugs. Next slide. There was also a separate investigator-initiated study looking at axitinib, a VEGF-based TKI with pembrolizumab. And in a cohort of patients enriched on this study, there was a 54% response rate. So we now have these drugs on our NCCN guidelines. And next slide. And recently, Alice Chen from the NCI did a study looking at atezolizumab as a single agent. And we see a 32% overall response rate in alveolar soft part sarcoma. This just recently led to FDA approval. So 3 independent investigator-initiated studies all gave us access to a drug. Next slide. But the question is, how do we use them? Again, small data sets not combined. We have all the drugs available, but we don't know. Do we start with the single agent PD-1 inhibitor? Do we start with PD-1 CTLA-4? Do we start with a PD-1 plus a VEGF TKI? Do we sequence them? What do we do if patients become resistant? But they're being used now in this patient population all the time in the United States, so the data is there. Next slide. So we have to learn from what we're doing with our patients. Every patient that we treat with an ultra-rare sarcoma, we need to collect the data prospectively, and we need to learn about it. Next slide. So this is beginning to, again, change our paradigm in how we approach the diseases. We are good at scientific discovery. We are actually really good at early signal finding studies. What we need to do better is to understand the natural history of these diseases, the clinical and genomic variants the patients need, so we can appropriately design studies for them. And then importantly, when we design prospective studies, we have to be able to have counterfactual and the prospective data to allow those studies to actually influence appropriate usage of the therapies, right? And that is critical so that we can actually tell patients, not only do we have drugs, but we know how to use them. Next slide, please. So the data is out there. In Europe, many of these diseases come to very specific centers, and I just have France up there that has a very, very well-defined sarcoma collaborative network, and they have really strong data. Next slide. So when they want to collect data within all of their patients, they can actually come up with accurate and complete histories. Next slide. In the United States, it's much more difficult because of our insurance processes. Patients go all over for their care. So they may actually be able to get a diagnosis at one institution and get a treatment plan. Then they go back home into the community and they receive that treatment plan. They need surgery, so they go to another academic center, or they need a clinical trial and they go yet to another academic center. Our data is fragmented. Next slide. We have to think about ways that we can pull all of this data in today. And just as we heard beautifully with Gerard, there's actually a lot of patient-reported data, self-reported out there as well. We need to incorporate that. Next slide. The United States just passed what was known as the Health Information Exchanges Act, so that if a patient signs a consent, we can now go into every one of their charts and we can begin to bring that data collectively into a common place so we can actually evaluate that. And we need to do that with all of these investigator-initiated trials that are beginning to show outcomes in small cohorts of diseases. Next slide. And so this is where we're coming in with this push platform, because this is the next iteration of our community. And when we speak of our community, we are speaking collectively of all the people within this room today. This is where we want to be able to identify the questions, the diseases, and then bring comprehensive data for evaluation. This is the counterfactual data that we need. Next slide. And this is just a depiction of all the types of data that we can actually collect. And this is powerful because this is not only natural history, it's outcomes data, it's genomics data, and it's all sitting there, right? So we need efforts that are beginning to collect this, and this is what we are trying to do. Next slide. But we need a prospective component to this as well. We heard that in our discussions with you. And so we need to think about ways that we can gather prospective data on every patient that is being treated. So as part of the PUSH platform, which is agreement with academic centers now around the world, what we want to be able to do is create roadmaps for all of these diseases so that if a patient comes with a rare disease, they can be treated along a very specific roadmap. Next slide. So if we create working groups, and one of those working groups is an epithelioid sarcoma, and we say we want to create a treatment roadmap so we can think about inclusion-exclusion criteria, the drug, what doses of the drug, when are we doing labs and radiographics assessments, how are we looking at AEs, how are we incorporating patient-reported outcomes, that if a patient gets treated at a site, they can enroll onto this and collect the data at their institution. So next slide. And so if a patient uses tazemetostat in certain sites, we can actually begin to collect that data prospectively. Next slide. If they don't have tazemetostat and maybe they're being treated with conventional chemotherapy, we can collect that data to create historical control data. Next slide. And if they don't have therapies, or if they choose to just observe, we can collect that data again in a uniformed way. So this is what we're beginning to try to do within the international sarcoma community. Next slide. And when we think about alveolar soft part sarcoma, with the several types of treatment that we have available, next slide, we can begin to collect this prospectively. So if a patient gets a single agent PD-1 inhibitor, or if a patient gets a VEGF TKI, we can see how they do. Next slide. And if they progress, and if they get something added to that therapy, or they get changed to the next therapy, we will have the roadmaps for them, and we can coordinate the data collection so we can understand outcomes with these data. So it's an opportunity to learn from every patient. Next slide. So why is that important? Because we are only just developing that, and we need regulatory input to understand if we can develop it correctly. We need to be able to work with you to say, as this data comes in, is it meaningful? Do we need to change the processes? We want to be able to help each other iterate on our internal efforts and our internal processes, right? Because I think it's going to be the general global input that's going to allow us all to come with a common data set where we can all use. So thank you very much for having us here.
Yes, thank you very much, William. I think you illustrated with several examples how important it is to find the right balance between the urgency of treating patients, but also getting sufficient information at the start with having data on natural history of the disease, information on genetic differences. And I think having a good methodology like you describe as a treatment roadmap is certainly something that is very important to make sure that whenever data are collected in a prospective manner, then it's actually the right data that we need as regulators. So yeah, thank you very much for this presentation. And the next presentation is by Lorenzo D'Ambrosio, who is the head of Department of Oncology in the University of Turin in Italy. And Lorenzo will talk about the development of new criteria for response assessment, and will use a case example of epithelioid hemangioendothelioma. So yes, Lorenzo, over to you.
Thank you very much. I have to disclose that I'm not the head of the Department of Oncology. But not yet. We are working on it.
You just received a promotion.
Right. It's always nice. Anyhow, let's see whether we manage to. The aim is basically to try to find a way to improve the response assessment that we already heard during this morning. That is a major challenge, especially in sarcomas, in rare tumors, especially when RECIST basically do not mirror what is the clinical effect that we see every day in the clinic, and when we cannot use randomized controlled trials because of rarity. Okay. I will start presenting these two situations, and I think that from a clinical perspective, we can all agree that these two clinical scenarios clearly depict an activity of a drug. Okay, the problem is that on the left-hand side, you have the patient 0 treated with imatinib. Thank you very much. And on the right hand, you have an EHE patient of mine treated with off-label sirolimus with an impressive metabolic response after a few weeks of treatment with the drug. So from a clinician perspective, these are more or less unequivocal signs of activity. And so we can also argue that we have to, in some way, to demonstrate what is obvious. But of course, it's a different matter if you sit on the table of a regulatory one. Okay. So basically, we have to make a small step back, and Robin already showed you this data on sirolimus activity in the retrospective data collected by Sylvia. But what I wanted to highlight here is that we have a quite huge discrepancy between what we see in terms of response rate, about 1 out of 10 patients achieving a partial response according to RECIST, and on the other side, more than 50% of patients achieving a disease stabilization longer than 1 year. So we have this kind of discrepancy. And this is in part related to the fact that we use RECIST, that are the best criteria we have in medical oncology, but of course they are not perfect for all the cases. And you all know that the RECIST are basically looking at the sum of longest diameters of up to 5 lesions. And they are quite good in discriminating what is working, shrinkage more than 30%, and what is not working, an increase in the sum of longest diameters of more than 20%. But sometimes the clues are in between these two thresholds. And so trying to improve this kind of evaluation through the years, several sons, let's say, of RECIST has been developed, starting by Choi criteria in 2007, then PERSIST criteria for PET response evaluation, and also more recently the immune-related response criteria. Okay, these are all signs of the need of improving RECIST in some situations. And no matter the criteria you are using, sometimes in EHE, we are facing a situation like this, and it might be challenging to choose the right 5 lesions to demonstrate activity of a drug. you might probably miss the lesion that is shrinking more. But this might be a topic of other discussions. And trying to summarize the challenge of response assessment in STS, we can for sure say that sarcomas may not shrink as other epithelial tumors. And this is because they can become fibrotic, cystic, or myxoid without significant changes in the size. And these challenges in evaluating response is also depicted by our substantial inability to really predict the activity of a drug in terms of response. We are quite good, and Alex did a lot of work on this, in predicting survival outcomes, but we are not good in predicting response in terms of dimensional variations. And this is also pretty much well depicted in the last paper on a phase 2 study in epithelial demand endothelioma with stromatinib that basically was a negative trial. But if you look at the waterfall plots, you can see that almost more or less all but few patients responded in some way with some disease shrinkage. So we also learn from the GIST story that shrinkage is not always what we need, and also that disease stabilization might in some way mirror the activity of a drug. So coming back to the HE, we were in a need. We were in a need because we are in a need, because we have no drug registered for use in Europe for this disease. But in the end, we have several data coming from clinical experience of the activity of, by instance, sirolimus used as off-label. And so we mentioned, and Pierre said, okay, retrospective data can be used to, let's say, raise hypotheses that should be confirmed. And so we need to confirm them prospectively. But the disease is too rare to randomize, so we have to look at single-arm trials. But single-arm trials' preferred endpoints are response rate and biomarkers. In EHE, we have some interesting biomarkers like GDF15 that has already been mentioned, but in terms of response rate, we are not so good in it. But the drug indeed remains active. So we basically came to the end that we need new response criteria to evaluate activity in EHE. This is just a logical syllogism, okay? And in this nice review from the RECIST working party, our colleagues pointed out some of the challenges that a researcher might face when introducing new endpoint validation criteria. And the picture on the right hand basically depicts the fact that when you introduce a new criteria, you are pushed on opposite directions. On one side, you can have specificity. On the other side, you can have generalizability. On one side, you have what you need in the clinical practice, and on the other one, what you need in clinical trials, and so forth. So in EHE, we came back to what we see every day in the clinic, and this might be a situation like this, where you have a diffuse pleural involvement, a pleural effusion, without a huge amount of measurable disease by RECIST. And so one may argue, okay, pleural effusion is not included in RECIST evaluation because it is not relevant. But we indeed know that in EHE, serosal effusion are relevant, because you can see here, once again from the Silvia's paper, that having a pleural effusion has a paramount importance in terms of a worse prognosis for patients. And the survival curves clearly show that you can move from a 4-month PFS to a 4-year PFS. And so if we do agree that pleural effusion is something that is worth to be measured, now we have to consider how can we measure pleural effusion. Of course, linear measurement is not the best one. So discussing with radiologists, we came to the end that we can measure pleural effusion by volume evaluation. And nowadays, with some tools available also as an open access, I would say, in the web, you can easily measure the volume of pleural effusion and monitor it. And so we develop, and we are here also to propose and to discuss these new criteria for EHE that we called rescore criteria, thanks to the idea of PANS that is amazing in finding acronyms. And we propose to add the serosal effusion evaluation to RECIST criteria. Basically, CR, complete response, is quite easy. So you have a CR according to RECIST and no pleural effusion. The PR is a bit more complex. You have one situation where you can have RECIST PR satisfied and no worsening of pleural effusion, or you can have a shrinkage of the dimensional evaluable disease by RECIST, once again, that is in between 10 to 30%, but you have a quite huge reduction in serosal effusion by more than 66%. Of course, this is applicable to patients who have actually serosal effusion. Stable disease is what stands between progressive disease and partial response as always, and we propose to add the evaluation once again of serosal effusion to progressive disease label. So you can have, of course, progressive disease by RECIST as always, but you can have also progressive disease in case you have a stable disease by RECIST, but a progression of more than 40% in serosal effusion. And now I would like just to show you a couple of cases where this might be applicable, okay? This is a case of the patient you already saw before. You have a baseline situation that you already saw with some pleural thickening, some serosal effusion. After 4 months of treatment, the patient started feeling bad with symptoms worsening. And a CT scan, you can see that there is a slight increase in serosal effusion in the left lung and the appearance of serosal effusion in the right one. And you can see the serosal effusion depicted by green as baseline and red after 4 months. And this was indeed a stable disease according to the RECIST, but the patient did worse. On the opposite, we had a patient with a baseline pleural effusion that it was FDG avid, that after a few weeks had a pretty much good shrinkage of pleural effusion. Once again, this is a stable disease according to RECIST, but might be a partial response according to new criteria. And what is interesting is that this was accompanied by a pretty good symptoms improvement by the patient, not only in terms of improvement in breath, but also in terms of improvement in general well-being. And one other interesting thing to me is that if you have a look at the PET scan, sorry, it is a bit slow, the serosal effusion was once again FDG avid and became completely FDG negative after a few weeks. Once again, mirroring the activity of the drug. Of course, we all know that in EHE, it is not only related to what we see in terms of radiological evaluation that matters, but also pain is a big problem for patients. And measuring pain is always challenging because we have a component that is completely subjective, of course, and you have something that you can measure in terms of analgesic use. So in collaboration with palliative care unit, we developed this pain response criteria, relying on worst pain intensity on one side and on analgesic dose use on the other side. And you can see here, once again, the criteria we are proposing. With complete response, that is absence of pain without variation in analgesic dose. Partial response is decrease in WPI score without changing of analgesic dose, or the possibility to reduce the analgesic dose. Stable disease, once again, is what is in between, and progressive disease is worsening of pain. And in the end, the last things we are working on in EHE is to try to improve the quality of life evaluation. All the patients enrolled in the already ongoing observational studies will receive both the EORTC and the ESAS-R questionnaires, but we are working with both the EORTC quality of life group as well as with the EHE advocates to develop new specific EHE criteria for evaluating quality of life. So, in the end, if you look at RESCORE criteria and at the endpoint validation criteria pointed out by RECIST working group, we can see that in some way we do believe that they sound biologically rational. Of course, we have to work and to improve the standardized protocol for measurement. We are perfectly aware of the potential limitations, and we do believe that in some way these criteria might correlate with the true patient benefit endpoint. Of course, usually this last point requires a huge amount of data that we cannot have in EHE, but once again, is a matter of what Keith said, so it's a matter of the grade of the degree of uncertainty that we would like to accept. So my presentation will end with two questions for the group. The first is whether we do believe that risk score criteria are acceptable or might be acceptable for EMA and FDA to evaluate the drug activity in EHE. And the second one is whether we can consider to have meetings like this one to discuss and develop together response criteria, new response criteria when we cannot use RECIST. So thank you very much.
Sorry, if I may add a comment, because thank you, Lorenzo. I want to say and acknowledge that this is, to me, the perfect example of how working together we can improve, because now Denise will present about the repurposing project pilot, but this idea of improving response assessment in EHE is the result from our side of researchers of the discussion we had with EMA about how we can show, how we can confirm retrospective data that are very relevant with some additional prospective data to corroborate, to be more convincing that the drug is working. So we have a room where we will be able to discuss rescore. It's not going to be done today, and this is what we are going to do in the next scientific advice, or I don't know how to call it now, repurposing of sirolimus in EHE. So this is part of the application that we are going to make by middle June. So we are lucky in that case because we have this option. What if a similar question, a similar issue has to be answered with new criteria in another ultra-rare sarcoma or ultra-rare cancer? Can we make use of a similar kind of meeting to talk about this together? So this is our question. Of course, today we don't want to go into detail on rescore, even though for us are very interesting, of course.
Thank you. Maybe we can take this a bit in the discussion, but certainly what it tells me is that at least it's a way to take into account what we said this morning, the entirety of the observation and the entirety of the data. So beyond the methodology, at least I think there is this aspect. Now it's my pleasure to introduce Dennis Robinson, who is joining us virtually, who is going to speak about repurposing and presenting the case example of sirolimus in epithelioid hemangioendothelioma. So Denise, if you are online, the floor is yours.
Hello, everyone. I am sorry that I'm not with you. I'm joining you from New York here today, and I'm grateful to the EMA and the EORTC for hosting this workshop. I want to talk to you, as Sylvia just said, about repurposing a case example of sirolimus in EHE. Next slide, please. So the EHE community is very fortunate to have been chosen to be part of this drug repurposing, the EMA's drug repurposing pilot initiative. And we're glad that sirolimus has been chosen. It's fortunate that we are able to sit, we as advocates are able to sit in the room with regulators, with academia, clinicians, other nonprofit organizations who are grateful to giving their time also to help our disease. And importantly, we recognize that there's one stakeholder that's not present at this discussion, and that is an unfortunate challenge that we face, and one of the things that we hope as advocates that we can figure out how that works into this framework as well. We're glad to be able to support the bringing forward of preclinical data, of safety data, retrospective data, and importantly, real-world data to this effort. Next slide, please. So as an advocate, it's important for me to say, and many of you have pointed this out today previously and well discussed, the challenges for people diagnosed with epithelioid hemangioendothelioma are not unlike other ultra-rare sarcomas. There are existing drugs that can meet urgent medical needs. Lives are on the line every day. Every day we talk to patients who need a treatment and they cannot get a good treatment. As we all know, the drug developer has little or no interest, absent incentives or profits. And I point out, again, you all know this, the current regulatory processes are a hindrance. They're not designed for small populations like us. And we're beyond a small population. We're an ultra rare population with a high variability in the disease presentation and knowledge of how to treat that disease. And today, no organization is accountable really to ensure that the drugs that exist that are well proven are used to treat every person that they possibly can. And this is a challenge that we as patients advocates find to be not reasonable. Next slide, please. So how can we improve this situation? What data can we use? This is the point of this discussion, and we want to keep having this discussion. What study designs are suitable? And importantly, from my chair, what roles can patients and advocates play? Next slide, please. So just a little background. I think you all know this on EHE. It's an ultra-rare sarcoma affecting less than 1 in a million people. Very dispersed population globally. High variability in this disease presentation. So we know that people are presenting asymptomatically with indolent disease. Some are diagnosed and they have slowly progressing disease right off the bat, and then highly aggressive, widespread metastatic disease. And there are some prognostic indicators, but very few. And if you're a patient and you're diagnosed with this disease today, and you are the indolent patient that's diagnosed, no one can tell you when or why or how to really assess when your disease is changing. Because many patients are seeing people who are not familiar with the nuances of this disease. So some of those indicators that experts are familiar with, community-based oncologists or primary care doctors that patients are seeing are not familiar with that. So this is a big challenge for patients. We know that there are an estimated 180 new cases in the EU diagnosed each year. And it's estimated also that over 50% of those require systemic therapy or some interventional treatment. But these are too few patients for traditional treatment trials. We know from data that we're gathering that the interventions that patients are receiving varies widely around the world, and again, with the disease presentation. So ultimately, we don't have enough patients to stand up traditional trial designs. So drug repurposing is a good case here for developing treatments for ultra-rare sarcomas and indeed rare cancers. Drug repurposing really does meet an unmet medical need for these patients, and it requires an appropriate plan in this dialogue where we want to find ways to find treatments for these patients to really push forward with drug repurposing. And I would say, I would advocate that ultra-rare sarcomas can't conform to common research, common cancer research. All supporting evidence. It was said earlier by Kit that all data should be considered. I believe strongly that the expert clinician scientists that we trust and rely on to help guide treatment for our patients should have a higher value. Their opinion should be of high value when considering the treatment development, not just the traditional data that might be available for common cancers. Next slide, please. So we've already seen some of this evidence for sirolimus and EHE. We have retrospective data. We have a case report published. There is a small clinical trial series. And yes, this is a small group of patients, but the data is consistent. Here in this table of 46 evaluable EHE patients, overwhelmingly, so 85% of those patients collectively have a positive response to the drug. And no other drug in this disease has demonstrated this effect. So, we want to look forward at what other data is needed and can be used Importantly, data is in progress with an observational prospective clinical registry that also is a little outside of the normal box for use. And then prospective observational study data, as mentioned, those patients are accruing. We'd like to continue that, but that data needs to be used to help build this evidence. Next slide, please. So we're thankful as a community that that data has been used and experts convened. Over 80 experts from around the world came together to define evidence-based best practices for an optimal approach to primary and metastatic EHE. And this is important because every single patient that we come in contact with, we point them directly to this language. We share with them this paper, we point them directly to this language. When they have questions, we ask them to take it to their doctor to inform their doctor if they're not familiar with EHE. So these experts published this data in 2021, and this opinion says that mTOR inhibitors such as sirolimus represent the preferred treatment options for patients with advanced and moderately progressive disease based on the small data that exists today. So we think that this is really important. We don't think that it is appropriate that this information and the opinion of these experts be left on the table and not highly considered in the treatment development for ultra rare and rare cancers. Next slide, please. So I want to share with you kind of a new type of data that's not been discussed here today. I love what Gerard was discussing from SPAN's efforts to look at what data patients are reporting outside of the clinical setting. This is patient-led research. The EHE Global Patient Registry is an ethically approved registry platform that is, it exists on the National Organization of Rare Disorders platform. That's a group in the United States. They developed a registry platform in collaboration with guidance from the FDA on how to collect structured data from patients to learn more about these diseases. So this is not meant to be a data analysis. These are snapshots from the registry. This registry has only been in existence for 12 months, just at 12 months this month. We have 220 participants enrolled. And this is some of the data and information that we have from those patients reported at baseline, which is when they join the registry. And then we also collect historical data. So we look at, we ask patients about their disease presentation, what signs or symptoms they had when their disease presented, and then ongoing longitudinally, we ask that information, what treatments they've had, and what their reported outcomes are from those treatments, which we think is very important. Those signs and symptoms and those patient-reported outcomes sometimes vary from what we might find in clinical notes. Sometimes that information isn't in a clinical note at all if we want to go and look at those notes. So you can see here that over 60% of the patients have reported that they've had an intervention. And then we look at 20% of those patients who have had an intervention said that they have had a systemic therapy. About 13% said that they've had an immunosuppressant. So that is important. But look at the variability of the treatment. So if we look at 220 participants over 25 countries, and we think about dissecting those patients down into these small groups of people and those different types of treatment, we have very few numbers with which to work if we were to stand up clinical trials. So again, this is patient-reported data. We know that we don't have every EHE patient in here as of yet, but look at the evidence here that sirolimus is the overwhelmingly leader in what treatment has been prescribed and used among patients. Now we want to do a data analysis to see what patients have reported as their outcomes and if there are signs and symptoms, changes and improvements, because we think that that will be important as well. Again, this is hypothesis generating data. That is part of the objective of this registry. It's not meant to be, right now submitted to a regulatory agency for consideration of a drug approval, but we want clinician scientists to take this data and investigate further. Next slide, please. So this is another type of data. So these are results from a structured survey that was initiated from the EHE community in early last year. We sought out to survey the global patient population to understand their perspectives and experiences with sirolimus for EHE. And we asked people who had been treated, we asked everyone who'd had any knowledge of, perception of sirolimus for EHE and their treatments. So 32 people reported back out of the total survey of over 130 people that they had ever been treated or were currently being treated. And importantly, we look here, again, these data, they're different types of data, but they all point to the same conclusion that sirolimus does have a positive effect and result for patients with EHE. So of the 32 people reporting, 66% of those people have had a duration of treatment over than 1 year, with 5 people having been treated for over 5 years. That's really important data to look at. We think that that points to a successful treatment for EHE patients. And then if you look over at the effect of sirolimus on EHE patients, 62% of patients report positive response to sirolimus. No other treatment has this reported effect by patients, and that is very important. They either reported that their EHE was stabilized or they had a good treatment effect. Their tumor shrank or stopped growing, and that is really, really important for this patient population. Next slide. So here I just wanted to illustrate again just the global disbursement of these patients. So that survey data that I just showed you, those 32 patients came from a few countries, and you can look at this here. So if we want to go out and we want to find these people and we want to enroll them in randomized clinical trials, or we want to do traditional treatment trials, it's going to be quite difficult. In this particular survey, we had 12 people from the United States. We have a handful from Italy and Canada as well, which all of those, every one of those patients' experiences are important. And we should harness that data, that real world data, and bring that forward. Next slide, please. So I asked the question, is sirolimus for EHE a good case for representing ultra-rare sarcomas for drug repurposing? And I would say yes. And now can we align on the types of data needed? Next slide. So why is this a good case for repurposing this drug in EHE? Well, I would say, again, from the Patients and Advocates Chair, a majority of patients report achieving disease stability or reestablishing stability, and that is from multiple data sources. And we believe that that is important. In some cases, over years of time, again, many of these patients, when they are diagnosed, they are literally not given years of time. They're told to go home and get their affairs in order. So to have that happen for years of time and have these patients on this drug is very important. Expert clinicians agree that stabilizing EHE is currently the primary target for treatment. We rely on that. We point to that to patients. Patients and clinicians accept that this drug will not work in all cases. We clearly accept that, but every single person that it can help matters today. The drug is well understood, it's well established, and this drug the approval of this drug for repurposing in EHE would meet an urgent unmet medical need and improve more patients' lives. And I have to just underscore here, patients who are diagnosed with an incurable cancer that does not have an approved treatment should not face barriers of commercialism or impractical or common cancer study designs when a clear and present opportunity to treat them exists in a real world setting. Next slide. So we would encourage you, again, as advocates, accept the limitations of ultra-rare sarcoma and rare disease populations, use all reliable and available data to bring treatments to patients. And I don't want to suggest that we undermine scientific rigor. I believe also that we should have reliable data that is ethically generated to the best of our ability. We as patients we'll bring forward supporting data to that. We should elevate the opinion of clinician scientists, experts in the field to assess what their opinion is of these drugs. So that rubric, the hierarchy of data evaluation, we feel that these clinicians should be elevated in that. And of course, illuminate patients' participations in research. This is important, to attract industry as well. Lack of patient accrual, lack of patient availability, being able to identify qualified patients has been a long time challenge in clinical research, and we believe that we can break down those barriers. We hope that this process and these discussions will continue to establish an inclusive process that requires all stakeholders. Even if each stakeholder can't participate actively, observation is important to understand that the processes are working. That is part of the objectives of the pilot process. We like that, and we want to make sure that all stakeholders are present. And define a plan, forward for ultra-rare diseases and ultra-rare sarcomas. Harness all evidence. I'm going to point back to Kit. I appreciate that he said that earlier. Consider all data. Don't discard any data. And I believe that is my last slide. Next slide, perhaps. Thank you. Yes, last slide. Really, on behalf of EHE patients, I appreciate that EHE has been represented here today. Again, I thank my fellow advocates who are around the table for allowing us to talk. Thank you.
Thank you very much, Denise. I think you clearly highlighted the difficulties and challenges of this product in this indication. Maybe in the interest of time, and yes, the next speaker, Gauthier Bush, I think you are Director of Clinical Research of the Anti-Cancer Fund. So yes, Gauthier, over to you. Thank you very much.
Yeah, thank you very much for the kind introduction. So I will try to manage my slides on my own because we were provided with a new tool from the EMA toolbox that seems to respond better to our needs. So thank you so much, Francesco in particular. So my role today and my talk will be about trying to learn or improve at least on how to engage with companies as academic groups, especially for ultra-rare tumors. So I think companies know how to engage with academic groups when they want to. I think it's much more difficult, especially in the context of rare tumors, on us academic groups leading and try to engage companies. So I'll try to do my best to suggest potential solutions to improve this. So yeah, if you look at that list, it's coming from Sylvia's presentation from the previous workshop, but the approvals in ultra-rare sarcoma. So you see that quite short list with EMA and FDA approval in the middle. As you can see at the bottom of that table, you have 4 drugs that were actually approved for the first time in some ultra-rare sarcoma. So they entered the market as ultra-rare sarcoma drugs, and they were developed by companies that you can see. Now they might find other indications in the future, which would be great. And at the top of the slide, you can see 2 drugs that were already on the market for other indication, and that were repurposed to some ultra-rare sarcoma. And as you can see at the end of those lines, then the sponsor of those trials were academic groups, so the Children's Oncology Group in the US and the NCI in the US as well for those 2. So this new indication was actually led not by the company, but was led by a collaborative group with engagement from the company, because it led to approval at least for the 2 of them in the at the FDA, and for 1 of them at the EMA level. So I think it's good to look at this and try to understand why that has worked and why in other circumstances it's been hard to engage with pharmaceutical companies. So we together with some EMA representative, with patient advocates, the Anti-Cancer Fund, and Sylvia and other EORTC sarcoma experts, we've put together a paper on trying to look at hurdles for ultra-rare cancers in general. And so, of course, we've talked already about all of this today. What are the main issues? And we group them into 3 categories. The first one, we can't do anything about this. It's the rarity of the disease. And so we've talked a lot about that. And one thing which is quite interesting and important, I think, is the limited patient involvement. So we're very lucky to have many people here, especially from the EHE community. But I think for other ultra-rare sarcoma, this is very difficult to get support from patient, and actually put a little pressure, I think, on the community in general, on the pharma companies as well, to move forward. So that's an important point. The other thing I want to highlight is in the second category about the drug manufacturer's position, and especially the last entry on the uncoordinated strategy from both companies and academic trialists. And we talked about alveolar soft pore sarcoma, and in the paper here we've listed all the anti-PD-1 trials in ASPS, and I think it was 9 of them with 5 different compounds that were conducted for a total of 130 something patients. And I think it's a bit of I think it's important to learn from these experiences that we could probably do better with a coordinated strategy. Then the last thing I want to mention, so in the last category about academic trials in regulatory submission, is the fact that indeed when there is data generated by an academic group, it's pretty hard to transfer that to a drug company for actually an extension of indication. And even then, if that doesn't work, you might want to actually submit yourself the data to the medicines agency, but actually only the license holder, the market authorization holder, can do that. So we're a bit in a vicious circle where it's very hard to actually show that data to the medicines agency. So this is what I've tried to put on this figure. So when you have a product that's already in the market and there is clinical data being generated, you can be into a situation, either that clinical data has been generated by the marketing authorization holder, and then the process is very well defined. And if they want to, then they will take it to the medicines agency. There will be an evaluation. And in the end, that might end up on the label in Europe, for example, with a type 2 variation. If it's not good enough, then it will be declined. When the data has been generated by an academic group, then, as I said, you need to have an uptake by the marketing authorization holder. So you, as an academic group, you depend on that decision actually to move forward. And so I'm going to zoom in on this situation, because for the first one that I just mentioned, okay, it can happen, but it's actually, I think it matches the rarity of the disease. It's a ultra rare situation. So in most instances, we'll be in that situation where the data has been generated by an academic group. And so I'm going to look at the different scenarios here. So first situation is that indeed there will be a sort of uptake of the data, but by a marketing authorization holder. That can happen. It's possible, though it's rare, especially when the product is generic and when the new indication is ultra rare. Here you can find some positive example of ultra rare tumors that were on my first slide on crizotinib and atezolizumab, leading to approval either by EMA or by FDA. But then there's the more frequent scenario where actually you have generated data, but there's no uptake by a marketing authorization holder. This is a bit the default when the product is generic, because you have many manufacturers and it becomes very complicated to engage with any. So there are multiple examples here in ultra-rare tumors. We talked about sirolimus for EHE. I know there's anti-PD-1 for gestational trophoblastic neoplasia is considered in chemorefractory GTN, is considered the standard of care, but no drug is approved for that indication, so it's used off-label. Very strong data for bortezomib for T-cell lymphoblastic lymphoma, same situation. And another example is sunitinib for pheochromocytoma and paraganglioma. So we have a list of examples in ultra-rare tumors where this is the main scenario. And so in the end, because there's no formal evaluation, we don't know if it's good or no good. Then it's up to the guidelines and the clinician to decide, but there's no medicines agency actually looking at that. And so what happens is, I said, then of course there's a way to circumvent that, which is off-label, which come with some potential problems and issues. So now, how to avoid this situation? There are different time points, I think, where you can engage with pharmaceutical companies. So I've put here, a road. You start from the ID drug in specific ultra-rare cancer, and of course, you ideally there will be no roadblock. Of course, we know there will be, but you will end at the finish line with a formal approval of that drug in this new indication. I think ideally you would need to engage with the pharma company very early in the process and come to a sort of pre-agreement on what you're going to do together to pass, if all goes well, to pass the finish line. There's a second situation where you actually have no real pre-agreement, but you're still in a position to generate that data, and then you need to present that data to the marketing authorization holder and hope or argue for them to take it to submit a type 2 variation. And then there's an even later situation where actually you have no commitment from the marketing authorization holder, and then you would want the medicines agency to look at the data package, which might be a possibility in the future with Article 48 of the future EU legislation. So, of course, the best is this one, is when you can come with a pre-agreement. So when you have that discussion, you can immediately decide with the company where you want to go, whether the data and the sponsor will be the company, and then you can go to the left on that pathway, or then there could be an agreement where actually the company will agree that you take the sponsorship of the clinical trial data, but they will submit, and so you know the pathway as well. I'm trying to move further, but I guess I need to accept the... Yeah, sorry. Thank you. So there are examples where pre-agreement has been found. So this is not a rare cancer, ultra rare cancer, but it's a ultra rare situation still, and it's a cancer drug was approved for breast cancer. So alpelisib in PIK3CA related overgrowth syndrome. The development was initiated by an academic group and actually was then led by the company, and I think that's really the ideal scenario. The other situation is a very interesting paper, but it's not in a rare condition. But the paper is interesting because it really explain precisely how they've done that agreement. So between the UCL and AstraZeneca, with UCL acting as the trial sponsor and then AstraZeneca providing funding and drugs. There was initially a startup agreement, so they did not need to agree on everything from the beginning. But then as things developed, then there was the signature of a full collaboration agreement, and they went together for a joint pre-IND and a scientific advice at EMA, with full agreement on the protocol, data collection plan, operational aspect, and so on. So there's an agreement that if all goes well for that trial, then there will be use of all of this for a regulatory submission. Now for still, okay, can I get the next slide, please? I think yeah. Where was I? Sorry. There we go. So the idea for pre-agreement, there might be, especially in ultra-rare sarcoma, some need for strategic forums where you will have multiple stakeholders, and then based on the data already available on the science and so on and so forth, it could we need to answer that question. Is it a good lead from a patient and science perspective? But I think we need to be honest with ourselves, and if it's not a strong signal, then also a good decision is not to do anything with it. But then if it's a strong signal, then we need to prioritize and make a plan. Of course, the collaborative groups are already ready and prepared for that, but we need to streamline trials for companies to make it attractive. And then as trial funder, and so the Anti-Cancer Fund is a trial funder, we need to promote this type of pre-agreement. Actually, we can request pre-agreement, which is quite a demand, but I think that might be our role to push things forward. Then the second time point is a late one. It's when you want to promote the uptake of your data to a marketing authorization holder. So you can see that the idea would be to move from the left-hand side, the situation on the left-hand side, which is probably what's now with the size of the arrows, to move to the situation on the right-hand side. And to do that, there are many ideas on how to stimulate uptake by a marketing authorization holder, and most of the time it has to do with money. And you need to give incentives to the marketing authorization holder, so a reward, a prize, a voucher, some differential pricing, so they could get higher price for the rare indication, or then lower the upfront investment in submitting, so simplification, reduce fees, some support from the medicines agency, and so on and so forth. So that's good, but it does not address some of the other complex issues that needs to be addressed if we can move forward, which is some due diligence on the data quality, an agreement with the data owner, some agreement on the liability, who is liable for the quality of the data. And then from a company perspective, the work you put in this is a potential loss. So you have an opportunity cost. And then in many countries in Europe, we know that if you get a new indication, then there will be a negotiation on the price, and you will be, as a company, you will have to lower the price of your main indication, which is really problematic from a company perspective. Then the third time point is very late, and we would move again from the left-hand side to the right-hand side, where actually an academic group, a collaborative group, could submit the data directly to for evaluation by the medicines agency, and that might happen in the future thanks to Article 48. And I'm not going to comment on that or say anything about this because it's still in discussion, and everything will have to be defined in how this would work. So my final thoughts are that from a funder's perspective, but I would say from a patient's perspective, I think we need to be demanding. We should not accept academic trials that are not considering the end goal, which is to hopefully get a formal approval by a medicines agency. From the academia perspective, clearly there should be a change in the mindset, and that the ultimate goal should be the approval. So I know many of you here are really aware of that, but I don't think that's the case for everyone. We should not do trial for, of course, we do trial for access for patient, but we need to think of the long term, and we should not do them for fame or a career advancement only. Then there's a need to prioritize, especially having good timing. When you think of NTPD1 in ASPS, for example, there was a time when that was necessary to prioritize and do it together and collaborate. And then we need to be clear amongst ourselves about the expectation. And of course, that's been said several times as well. We need to educate ourselves about the regulation and drug development in general. From a company's perspective, I think we should push them to say, this is your responsibility to test drugs in rare cancers and population. I think we need to really push on that button, and from that angle, be really strong with that message. Not sure that's going to work, but I think we need to continue telling this. And then the companies are very good at having very high quality data. So I think we need to learn from that, and we need to ask them to help us to generate data that will be good enough for submission, which is a real concern from medicines agency perspective as well. And I think we know that the pediatric oncology community has worked on that with the concept of strategic forums, and I wonder if we should not put that forward, ultra-rare tumor strategic forums. It's happening here and there, but not at the international level, and I think it would be really nice maybe if we could test that and see whether it delivers or not. Thank you for your attention.
So now we move to the discussion of the second part. And I think I would like to start with perhaps summarizing what we have learned so far. And yes, if I try to make this summary, I think what I understood from all the discussions is that it's really important to have a plan, to organize. And I think if I want to try to do this, I think we should start with the identification of patients. And I heard that what is important when identifying the patient is baseline characteristics, the medical history, including the baseline treatment reported. We have also learned that we have existing social media platforms like Facebook, where patients actually get together, and that has been a very positive success story. And then I also learned that the exchange of information from patients could be facilitated by the Health Information Exchange Act in the US. And in Europe, we will have the European Health Data Space, which will greatly facilitate the exchange of information and information on the patient's data. We really need to have a collaboration and cooperation between academics so that there is an evidence generation plan, and especially when there are several perhaps products to be treated, to be tested to patients, we need to understand what has been done so far and build on the lessons learned from existing retrospective data. And really have a good evidence generation plan, including a rationale why a certain product could be selected to treat a specific group of patients. And we learn about the good lead from a patient and science perspective. I think that was in one of the presentation. And of course, it was also mentioned that we need to really understand the mechanism of action, which endpoints would be the best to be used, if PRO's data need to be collected as a safety evaluation, but also the follow-up of patients, and that could also be done using digital tools. So I think we also learned that there is an urgency, of course, to treat patients, and for that, we can use the compassionate use program. And we were already informed about the French case about the harmonization of data, and that could be further discussed and perhaps aligned across Europe. That could be a pilot. So it's always important, even if it's used as a compassionate use or off-label use, that there is a standardized way of collecting the data so that we can further use this data for registration purposes. Ideally, we want, and I think it's we have learned that we really would like to have prospective data collection with a standardized protocol for interpreting measurements. If possible, randomization, but we have also learned that with very small number of patients, it might not be always possible. And then, of course, to discuss specifically and for each indication, what would be the best endpoints to be used. We had a proposal about the risk score, which is more looking at symptoms improvement, and of course, quality of life data to be considered. Then at the very beginning, Pierre mentioned about the innovation task force and the tools so that we can get together and give advice to academics and trying to find new ways or existing ways, but adapt the existing tools we have, such as the Innovation Task Force and the protocol assistance. And we have already existing collaboration with FDA. So for us, it would be actually very envisageable to have this jointly with FDA. And of course, it would be very important to discuss the study design and endpoints before academics embark into such clinical trials. And that has to be done for each indication. We also discussed that this has to be disease specific. Yes, so I think once academics get feedback from regulators, this could help them in order to get funding, because at least they get feedback on what would be the regulators' expectations in terms of data. And then for them, they can always share information that we made available to them to facilitate further partnership whenever possible. So I think that's my summary, and I don't know, Denis, if yes, you would like also to further comment.
Yes, I try to put a little bit the example we heard this afternoon in the perspective of some what I believe were important messages this morning. I would like to refer, for instance, to the message of Robin Jones about off-label use, but also Andrew, what you told us in your presentation, moving from retrospective to prospective data. And I think the challenge that we have is that if we meet next year again or in 2 years, whatever, what I would like is that we no longer speak about retrospective data, because all the data would have been collected prospectively and would be of sufficient quality. So what do we decide today, because of the urgency, to change this paradigm? And I think indeed for this, we need to very well understand the battlefield. Because if you want, well, I'm French, so I inherited from Napoleon. If you want to win the battle, you have to know the battlefield. So what do we have? What do we have in Europe? And I would like to echo what the solution that exists. And again, what I already said this morning, we really need to really embrace the whole thing. Otherwise, it's not going to meet where Europe is going in particular. And William, you have put the French system. I think it's an important example. But for instance, in Europe, there is also a European infrastructure called Huracan. EORTC collaborates a lot with Huracan. We have done the ArcaGen project with them, and this is a sort of prospective infrastructure that already, I would say, achieved a lot of the things we have discussed today. So EORTC has already this solution that you may want to exploit. But the benefit also is that Europe likes to build on what it has achieved. And I think if we want to exploit the solution that Pierre has proposed with the Commission, building on what exists, like Huracan, ArcaGen, and so on, could be a substantial added value and the boost to what we can create from there, because then things come together in an environment that we know. So these are my ideas. Indeed, other things exist, European health data space. So how can we benefit of all that? We really cannot build things in isolation. Otherwise, it's going to be difficult. But again, I think important messages this morning. Thank you. So that would be my first reaction. Maybe you can invite the two.
Yes, I think we can perhaps invite also, maybe we start with Marta Donoghue, if you want also to share your views from an FDA point of view.
Hi, everyone. Thank you so much. I think I don't know how much more I can add from what's been already said, because I think from an FDA perspective, I think our sentiments pretty much align with those that have been expressed, particularly by EMA, but also by many of the other speakers, in terms of the kind of urgent unmet need to develop an infrastructure for more prospective data collection from every single patient who's receiving treatment, whether it be through off-label use, or through use of an investigational drug. And I think we do have a lot of ongoing work streams, but I think, as someone was just saying, sometimes a lot of these efforts are independent. So having a mechanism to coordinate these more would really be beneficial. I'm not personally that knowledgeable about Huracan, but I just looked at it and it sounds like a wonderful mechanism to help systematically collect data. And as I think somebody had mentioned earlier, I think the NCI also has ongoing efforts, particularly in the childhood cancer space as well, for more prospective data collection. And I think, I would only emphasize that, we would very much like to be a part of any effort that is currently underway or already ongoing that relates to collection of data systematically for, in the real world or otherwise for rare patients, because I do think that as the sentiment has already been expressed multiple times, the data from every single patient really matters. And, we very much want to make sure that we're leveraging that data, not only to benefit that patient, but all patients. So, I really appreciate being part of this discussion, on behalf of FDA, and really look forward to continuing these discussions. And, in terms of the retrospective versus prospective data collection, I 100% agree that the ideal is to have a prospective mechanism to capture this data. We have in the past used retrospectively collected data to support approvals, provided that it's then analyzed in a prospective, unbiased fashion. But, obviously the ideal would be to collect that data in more real time in order for it to be as contemporaneous as possible and useful as possible for regulatory purposes.
If I may, I want to spend a word in favor again of a retrospective, coming back to the idea that we need to take advantage of each single data we have. And this in particular is not to discriminate new entities. Again, in sarcomas, luckily or unfortunately, our pathologists identify a new entity at least every 6 months, meaning that we do not have the chance of having prospective data, for example, in this case. So what I mean is that I think we need first to collaborate all together, and each partner should put on the table the capability, the resources, and the willingness to work together. On the other side, there are different conditions. There are conditions that are known from years, and for example, the natural history is already known, the activity of conventional drugs is already known, and in that case, maybe it's important to really strongly go only for prospective data. But there are other conditions that are new or almost new. And in this case, we need to start generating hypotheses. We need to start thinking of what kind of prospective data we want to collect, looking at the retrospective data we have. So I think that is important that all together we define what are the criteria to be able to get all the data in a quality, with a quality criteria that can work. And this applies to all the kind of data we can collect: translational data, retrospective data, observational prospective data, prospective registry, and also interventional prospective study. Of course, ideally, the last is the best, but not always is feasible for the reason we have listed today and also the other day. So we must be concrete and think that a patient that is diagnosed with an entity today needs a treatment today, not in 10 years from now. So maybe in 10 years, we can build a prospective data collection and we will be able to offer a better treatment, the best treatment, and new drugs. But the patient is in the clinic today, and he wants to participate to, also the research, but still need the treatment today. And for that reason, we need to build on everything we have, every single data. And we need to align, to agree on what are the criteria to collect this data in a good way. All the data we can gather from patients, from databases in the clinical study, making available the data from clinical studies that often are not reachable. And this is another point that we didn't address today, but is really shaming to me, So we need to be open, not to close. We need to be open to make use, the best use, the most rigorous use of everything we can get, without being, focused only on something that is maybe the best when available. But if not available, we need to use something that is maybe not the best, but still relevant to our patients.
I see several hands.
Free. So
yeah.
Maybe you are first, second.
Yeah.
So maybe I can just add a little bit to what Sylvia just said, which is, I think it was in, maybe go to your slides, a list of drugs that have been authorized. But two of them on that list in particular, crizotinib and I'll say nab-sirolimus. The initial descriptions of the benefit of ALK inhibitors in inflammatory myofibroblastic tumors and of mTOR inhibitors in malignant PEComa were shown in 2010. That's when the original case reports were published. The drugs were authorized in the last couple of years. This is an example where there's a target in the disease. There are drugs that are approved for other indications, and companies were not interested in developing the drugs for those indications. So we went more than 10 years without approved drugs for those indications. In the US, of course, we could find ways to get the drug for many patients, although for some patients, insurance would still say no, they weren't going to cover it. In Europe, there are different situations. But here's an example for me where do we really need prospective studies to show that those drugs are active? There's a known target, case report demonstrating efficacy, known activity, known toxicity profile of the drugs, and no other defined treatments for those diseases. Just because pharma wasn't interested to invest a few $1 million more in developing the drug that would benefit 50 patients a year, we need a better way of getting there. And even if we did prospective observational studies, we still wouldn't be at the point until we did a prospective interventional study to get there, which took more than 10 years to do. So again, just an argument, I think, supporting what Sylvia said is, in some circumstances, we might have enough data, which is really based on case series or retrospective data.
Just to say, it's Robin Jones here. I agree, Andy, that's very well put. And I suppose that was the point I was trying to make earlier on today about using the totality of data available about a specific subtype. But I think you put that very well.
So I agree with all that, but I'm actually going to have a shameless plug. And Martha, I really appreciate what you said. And I just want to let you know the FDA has been an absolute wonderful partner to sarcomas and ultra rare diseases, not only in helping us get drugs to our patients, but also kind of the continual dialogue that we've had over the last few years and even through COVID. And I would love to take you up on the offer of kind of explaining more what the community's doing about data collection, because I think without the FDA's input, just like, we're trying to get the EMA's input, we may not be, on the right path. And since so much of this is still in conception and we're figuring it out, we would love to figure out how to partner with the FDA as well.
Yeah, Martha, would you like to?
Sure. I wanted to be sure I wasn't going to speak over someone else. No, I just reflecting on sort of the discussion that has just been taking place, the two things, and I'll get to a little bit of elaborating on ways to interact in a minute. But with regard to using, I really appreciate the crizotinib example in particular, particularly because I'm a pediatric oncologist and, thinking about the, timeline for approval of crizotinib for and ALCL, anaplastic large cell lymphoma as well, for those patients was a very long timeline. And, I think there were multiple reasons for that, but and again, I think going back to my comment about making sure data from every patient matters, I think, we are and have been and will continue to be flexible with respect to what the data source is to support an application. But the quality of the patient level data is really crucial to our review in most cases, and that we generally far prefer and for the most part need the granularity of the data being collected on the patients, in terms of their outcomes, rather than sort of a anecdotal case series without the availability to look at that data. So for us, most of the examples that I can think of where we've, used some retrospectively collected data, and there isn't a lot of examples of that, but generally speaking, the ability to go in and capture granular details about each patient and ensure that the patient sampling is not biased is very important to us. And so it is important to use patient data, and it can even be used as the primary basis to support approval in select circumstances. But generally speaking, we like to be involved as much as possible early on, if that is being contemplated, in order to make efficient use and kind of de-risk that approach overall. And we definitely welcome interactions with stakeholders who are contemplating that. With regard to potential ways to interact with us, in efforts that are ongoing or underway to construct registries, other types of systematic methods for data collection to support product development. We have, as you probably know, a fairly robust Oncology Center of Excellence real-world evidence program, and there are mechanisms to meet with us, even at the early stages, even before there's much work being done to discuss concepts, the types of data that we think, in terms of the data elements that would be important to capture. We could provide thoughts about data sources, et cetera. Typically, when we've interacted with sponsors, there are generally multiple conversations that need to be had because these plans are really detailed, and we need to be, thinking about, that the elements in a very specific way to ensure that we're not missing an opportunity to really fully leverage that data. So, if there are questions, I welcome people to email me and I can help direct, that process. What would be the right type of interaction and make sure that we can accommodate that. And I also think it would be great to do that in the context of having our European colleagues as part of that conversation as well, because obviously it's more helpful if we can provide some type of coordinated input.
Could I leap in there very quickly? Oh, sorry.
Oh, no, that's it.
Thank you, Martha. We have a lot of people wanted to give the floor. So yes, we have Pierre and Pan. Okay, Pan first, and then Kit, and then Pierre. Or Pierre and Kit.
Yeah. Today's been incredibly interesting, and we've talked about a lot of different things. We've talked about maximizing the use of data, so we leave no data behind in the same way that we leave no patient behind. We've talked about new sources of data from social media. We've got lots of questions about the best way of making our retrospective data more useful. These are still all open questions. And so we're grappling with these questions as we're trying to build push. And we heard from Pierre right at the beginning, very positive developments at the EMA. So how can we make this more concrete? Because doing this in an ad hoc way, it's a guarantee of not making progress quickly. So can we look at some way of formalizing the relationship so that we can discuss this regularly and actually have a timetable for deliverables? Because we've got so many questions on the table, we could be talking for years and not come to any concrete solutions. So my ask is, given the urgency that we've heard from many times during the day, what can we do to make things more concrete to get to where we want to be?
So many reactions to many points here. The first one, I'm afraid we are going into some misunderstanding. We all agree on the fact that all data available in very rare situations must be taken into account. It would be a pity that any data is neglected. At the end of the day, it could be very poor quality data, and we have good reasons not to use them. But we must use everything. So retrospective data are interesting, and exactly for the reasons you gave, Sylvia. It could inform us on the natural history of the disease. It could inform us on the activity. It could inform us on the mechanism of action, anything like that. So no one is saying that we do not want to pay attention to retrospective data. No one here. But in the not best case scenario, in the best case scenario, it could be enough, as I described before. But sometimes we are not in the best case scenario. Retrospective data indicate that the drug could do something, but to which extent? We have some symptoms that could get better, but we will never know whether this is really of an uncertain benefit to the patients. And we need more data. And here, it must be prospective. It must be prospective because it is what is more convincing. So we are all in agreement on that. And I believe that we already got some results because we opened the dialogue. The dialogue is open for the sirolimus in EHE. And this is something which is like a pilot. So we are not discussing where we are with that because the scientific procedure isn't going, but we are already in the discussions. Another point, I heard this afternoon some proposals for endpoints that could be more reliable and more relevant for a given indication. We have something which is called the qualification procedure, which is in the scientific advices, something that could be used for that. So if you come with a good idea of an endpoint that could be used in a specific situation, and you believe that this endpoint is mature for validation or anything to be exploratory, we can discuss. So is the volume of pleural effusion something that could be considered? So I have some preliminary remarks. We are not there, but okay, it can be discussed and offered as a qualification procedure. So we have the tools available already. I believe that we are in good agreement. We heard some examples. So I would like to avoid a situation where we go back home disappointed and saying nothing has changed and we are in disagreement. No, things changed already. We must discuss for any specific situation. We must find an agreement if possible. We must be prepared to the fact that sometimes we do not find an agreement. It could happen that you come convinced that the drug is useful to the patients and the CHMP will be of the opinion, and after all our discussions will be of the opinion that this is too uncertain to result in a marketing authorization. It could happen. Let's be prepared to that. Being open doesn't mean that we are accepting anything, but we will do our best to favor the positive issue whenever possible. So this is something, isn't it? We do not neglect any piece of information. We can offer collaboration and dialogue and build something that will be used in any similar situation to offer the best opportunity for a positive issue for the best of the patients. This is our preoccupation, and not being too slow on that. Okay. Just some good news. We have plenty of processes. Listening to Martha, we are in very good agreement. And you must know that for the scientific advices in the domain of cancer, we have every month discussions with our American colleagues just to check that we are on the same line. And if we are not, just to understand why. So every discussion we would have on these very specific and important points would be necessarily a discussion that wherever it begins, in the US or in Europe, it will be a bilateral discussion, probably. So don't be disappointed. We are prepared to offer our best. So this is exactly what I wanted to say to avoid the bit passionate the reactions to this afternoon. The last point, which is completely different. I remember having attended to one of the first meetings of Accelerate. And here I'm speaking to the patients and to the physicians. And we had 4 or 5 industries coming for very exceptional pediatric cancers, each one developing the drug and subtracting 10 patients here, 15 there, 12 elsewhere, and saying, oh, these patients are too exceptional, we cannot do better than a bunch of observations. And my thought was, but this is competition. It doesn't make sense. If we put all these people together, only one of them, and use the full available population of patients for collecting an evidence, instead of having 6, 5 very poor developments, we could have one very efficient development. So my message is, it is up to you, because we do not have any control on that. If you could help in protecting the future evidence from the competition, and if you could make your best efforts, instead of chasing the best publication ever, to offer the patients the best development ever, it would be marvelous. I was considering whether we could change the regulation to change from a competitive model to a collaborative model. So I gave up, it's much too complex. The floor is yours on that point. I count on you because I always find it, even in the scientific advices sometimes, we have parallel developments for nothing, because we must have 30% responses obtained in 6 patients. What do we do with that? But we have that repeated twice, four times for nothing. So this is completely different, but it is something I would like you to consider. It's very important, I think, to select the best one and to concentrate on that one.
Marta, you've raised your hands. You want to have a rapid reaction on that, but because we have other people waiting for speaking as well. So please be short. We can't hear you.
Thank you so much. I don't want to cut anybody short at all. I just, really, liked a lot of what I was hearing from you, Pierre, and I think that's very true. We just had a pediatric subcommittee of ODAC meeting where we were talking about prioritization of investigations for pediatric patients with cancer. And, what came out, a lot of the discussion was really that, the fact that we need to have a coordinated approach, because otherwise, with very rare diseases, you have too much duplicative, unnecessary studies, often, which are negative, and, the questions could be answered in a much quicker way if we had a more coordinated approach. I also just wanted to say briefly about the question about how to make concrete progress. And I think this kind of aligns with what Natalia just put in the chat, is I think in general, in interactions with us, if you can break down the problems in the process into concrete steps and questions, then it's a lot easier for us to provide, constructive feedback in general. Particularly for something that's very complex, like what we're talking about, whether it be, master protocols, platform trials, real world data collection, et cetera, as if you can organize kind of the project into smaller digestible pieces with, very discrete objectives and questions for us, then it makes it a lot easier for us to have meaningful interactions.
Thank you, Kit. You've been waiting for long. Sorry for that.
No problem. The only thing is I'm traveling back a bit to the discussion we had before Pierre brought a broader context, although I do think it's relevant, because we kind of had a discussion on the retrospective, prospective and what's better, but I would propose to frame the discussion differently. And it's actually, how do we get the right level of scientific rigor into our decision making, and how do we have the ability to get the uncertainty we have to the level that we're comfortable with? And then look at retrospective data. For me, the word prospective is still very relevant, also for retrospective data. I think it's totally different if I design a single-arm trial and I prospectively, before the trials even started, define how I intend to use any data outside of the trial. That's a totally different situation than coming up with a single-arm trial and with next to that a story on the additional data that has been collected. Now you have to come up for assessment. In the second situation, the uncertainties are almost impossible to address. In the first one, we can. So I would very much frame any discussion about data on what are the prospective research questions that we want to address, and how do we intend to use the data that's there or the data that's being prospectively collected. So that's kind of where I come from, and which I think is really important to move on, because otherwise we kind of stick into the dilemma with the randomized clinical trial as golden standard and no way to move. So I think this is crucially important. And the second point is some questions cannot be answered retrospectively, even not the good proposals we discussed today. If we are considering novel endpoints, that's usually difficult to address with retrospective data. At the same time, it could be actually very, very influential if we want to move ahead. So I think also that awareness needs to be there. And on the prospective data, you mentioned the qualification procedure. I think we have a dilemma there. We are in a very rare cancer, whereas if you do the traditional pathway, you have an independent validation of your endpoints. We might not have the luxury. We might have to make sure that we at least prospectively set a very good standard on how that endpoint is defined and data is collected, but are in a position that at the same time that we assess the trial, have to also assess, do we trust the endpoint that's used here? So I think, but that could be actually on the agenda on how do we do this type of changing our thinking about endpoints in such a rare disease setting. It's also not unique, happens in other rare diseases as well. But that could be an agenda topic. How do we actually want to do that? Because the traditional idea, we have a couple of years to validate an endpoint, and then we see it back in trials or in interventional studies, which also take a couple of years. That's probably not the luxury we have. So those two things for me are very much connected.
I think you're right, and I think we are open for dialogue. I think also, and that's what Pierre was saying, that we can have this more flexible way of meetings. So I don't know. I saw that Paolo Foggi from Scientific Advice Working Party had his raised hand. So I don't know, Paolo, if you also would like to comment.
Yes, very short. First of all, I tend to agree with Keith on the last comment on the qualification for the endpoint, maybe. But what I wanted to say is that I guess that I see two ways. One is to be concrete. Go into the specific cases and to look where the data are, what the kind of data we have, what is the maturity, what are the issues, and this should be done using, in my view, the tools we already have, and at the SOAP and the pilot of repurposing, and this should allow us to discuss concretely on cases and together see what we can put forward to the marketing authorization, or what is missing and what's not. And these are really cases on which we might learn. And then there is in parallel something on the planning in perspective. And again, we come back to the fact that we want ideally prospective data and ideally high quality data. And on all of this, I guess that we can find an alignment on the broader scope, long-term scope. So I guess that we have some issues on the table, hot, because the patients cannot wait, and there are some drugs that might be useful. So let's look at this example. Let's discuss together. Let's see what the limits are. Let's see how we can handle uses the standard tools that we have. And as Pierre said, we have also interaction with the FDA. So I guess that it is possible with these tools to get an alignment, to try to seek an alignment. And then prospectively look at the methodological quality data and so on. That's what I wanted to say.
Thank you, Paolo. I think the next one was Ralph, and then Hugh as well. You have been waiting. So maybe Ralph and Hugh then.
Two elements to that. All data. I think the regulatory thinking is to go from observation to general recommendation. And if we think of one sort of data being all existing evidence or retrospectively, then this could be a specific registry that is highly dedicated, curated, and so on. And I think this is in the hands of the clinical researchers, can be much tailored, it will be refined and so on. That's important. There, the regulators have the question, how can this be generalized? On the other hand, you have automated data collection, electronic health record systems, etc., that may offer to just have more and all data. But I think we would kindly ask you, please check, do you even have an ICD O code, for example, for EHE in the EHRs? Probably not, because you don't have it in ICD-10, and then you need to have something specific. So
what can you do to influence these type of things that are coming up together with the clinical support decision systems that will be prospectively relevant, because that is also beyond our reach. So that was the first element, that these are complementary data sources, maybe the EHR less biased, but the registry more granular. But what you then, in a second comment, can bring together is a plan for merging this evidence, for bringing it as part of a case for a medicine or so, or even for the natural history. And this argumentation, bringing it together in a case, in a research protocol, this is what can be discussed in a scientific advice. So the practical suggestion, I think I'm going out on a limb here, and colleagues, you will tell me, but we said before, let's have further general discussions on overarching topics, and then have case-specific discussions, and we need to see how these can be organized and how these can be enriched with perhaps manufacturers' insights and science, etc. But then there will be steps to be taken by the research group to go for a scientific advice, and then use the scientific advice to maybe take a next regulatory step. So these are the elements that I see: common discussion, breakout discussions, scientific advices, and then taking the data or research results forward. And we are happy to conduct several of these meetings in this year and next year.
Another workshop, no. No, I think you were next, patient's perspective.
Thank you. No, I was just, Pierre talked about efficiency and not running parallel processes, etc. This is a fantastic process we have running here with the EMA and the FDA. The thought of then having to do this with the UK regulator and the Australian regulator and the Canadian regulator, etc., fills me with dread. I don't want you to answer this question now, but one of the things I really would like people to think about is, can we develop this process to include those other regulators? And that, I think, has a massive impact on industry interest in engaging in this process, because if a pharma knows they can come to a process and we'll hit 6 of the big regulators in the world in one process, that may create great interest. So that was just the point I was going to make.
Yes, I think that's a good point, and maybe it's worth mentioning that we have monthly cluster meetings, so not only with FDA, but also with other agencies, which include PMDA, the Australian agency, Swiss Medic. So yes, and that kind of cases could be brought to these discussions, and hopefully for alignment. So that's already there, so it's existing. So yes, of course, that could be taking place. I think you were next, Gauthier.
Yeah, thank you. Pierre, you mentioned Accelerate, and actually I attended the past or the last Accelerate meeting, and I was sitting next to Martha. And so I have a question for Martha, because I realize you've been involved for quite a long time in this. And they organize those pediatric strategic forum as well. And this is one thing I was suggesting at the end of my talk, that maybe that could be a solution for some ultra-rare sarcoma. Let's say you pick one and then you organize a strategic forum on that to engage companies, to prioritize, to coordinate the effort prospectively. Then, based on your experience, do you think this is a good option for us, or maybe something else should be defined because of the specificities of ultra-rare sarcoma?
Is it okay if I answer? I don't know how many hands are up in the air. I thank you for the question, and I can only speak from my perspective about my experience with the Accelerate platform. I think overall it provides a really unique and important mechanism for freer exchange of information and discussion across multiple stakeholders, academia, pharma, regulators, advocates. And it's unique in that there is that kind of meeting of the minds and opportunity for free-flowing discussion among all of these stakeholder groups. And I have found it to be tremendously valuable. Those meetings are not decisional, so they don't we the regulators don't make, any, firm decisions on anything, but we do take that information that we're hearing back to all groups and use that information as part of our, decision-making process, particularly with respect at this point to the requirements for pediatric studies. And importantly, I think it also raises other questions for the entire group and potential projects and work streams that can be then taken forward in a working group method to help work on, some issues that are obstructing drug development. So yes, I think it's valuable. I think such a concept could be helpful in this space as part of, a sort of a more multidimensional kind of solution. But I do think the efforts are valuable.
I would like to elaborate a bit more on what Kit said, and thank you very much. I fully agree with you. I was just wondering whether, from a regulatory perspective, it might be foreseen that even criteria, no matter whether this risk score criteria that we are presenting here today, the first proposal or other criteria, might be considered, even if we will never have the possibility to validate in a standard fashion this criterion. So for a new criteria in ultra-rare diseases, we will have in some way to rely on clinical observations and in some way the biological rationale behind the criteria. So I know that it's probably a question without answer, but it's something that might be important for us to know, because otherwise it is hard to think of something like this. And then I have another question.
Francesco, yeah.
Your question is exactly related to the point for which I was asking the floor. We can use an endpoint in a given clinical development because we believe that this endpoint is the most suitable endpoint in the context of this drug for this disease. This is a scientific advice. We can find an agreement in the context of the scientific advice, but it will be restricted to the scientific advice, to this drug and to that disease. I was mentioning the possibility of having qualification. This was for the qualification of an endpoint that could be used beyond the specific development. Let's take the example of I have a drug which doesn't shrink the tumors, but that could offer stabilization of the disease over years. Why not? But this could be used for many developments once the endpoint is validated based on proper justification. Or I have a disease where I cannot expect tumor shrinkage just for the reasons you explained. Could I, on proper justification, another endpoint used in any similar situation upon justification? So if you have the question in the frame of a specific scientific advice for a specific disease, a specific drug, yes, the qualification process is not the right tool because it will take too long. I completely agree with that. It is impossible. But if your question related on the endpoint could be generalized to many more developments and many questions you're facing, not necessarily in the context of orphan diseases, it could be even for more frequent diseases, better coming for qualification. This is my answer.
Thank you very much. And the other,
Lorenzo.
Sorry.
Yeah, I'm always in this meeting full of admiration for all the courage really that patients, clinicians have in developing drug in these diseases, and I was, struck by your wisdom, but this time I was also struck by the regulator's wisdom, and Pierre at one point said something which I heard from your predecessor at the Oncology Working Party, which is, well, response of the disease actually might be a criterion which is on occasion sufficient to say this drug works. Okay. And if I couple with what Kit says, that uncertainty is not something that is an absolute regulatory threshold, but it's
an omnipresent thing when you look at data that we simply have to manage, and we have to decide in a collegial fashion what is acceptable uncertainty. So if we combine the two, I would say there is a reflection to be had here, because if the clinician and the patient tell us we can design criteria that are clinically meaningful, that tell us something about how the drug is working, how the patients are benefiting. And this doesn't require complicated randomized trials, but we trust a response when we see that it is meaningful, and the regulators are open to look at this. Well, that is a very important step forward. So I would say something we need to take home, Pierre and Kit, is, well, if there is a clear message from the community that there is clinical relevance, clinical value in a response, even an improved response as we have seen it, well, who are we to say that we need, surrogate validation or whatever? We should be able to value that and to consider that in the benefit risk assessment. So I think that is a very important step forward that we could do in taking this forward in the Oncology Working Party, in our guidelines. And one thing that Hugh said that struck me, he said, well, a rare cancer is not rare if you have it. It's just a frightening disease. And actually, Sylvia, another thing that we need to change is the title of this meeting, because you say rare sarcomas, ultra-rare sarcoma as a model for ultra-rare diseases. It's not just for ultra-rare diseases, it's a model for any disease with a very high unmet medical need. And so actually, what we are developing here has actually even wider implications for all those situations where you don't have drugs available, and where meaningful responses that clinicians and patients perceive as beneficial can be interpreted.
Yes, I think we had also several people online. So Marta again, and then Denise as well. So maybe Marta first and then Denise.
Thank you so much. I really appreciate the questions with respect to validation of different criteria for response and other endpoints. And I do agree with a lot of what I'm hearing here. I think in terms of discussions regarding any specific new criteria for use to support evidence of efficacy, I do really strongly encourage that parties request meetings with us, and maybe optimally collectively, so that we can discuss this these types of questions with our other regulatory partners, because certainly we are open to thinking about new endpoints. There are a variety of ways to go about validating endpoints, and I think a lot of that has to be contextualized. Are these endpoints going to be used as the primary efficacy endpoint? Are they going to be used as supportive information to help provide some confirmatory evidence? And there's more than one way to go about achieving validation. And so I do strongly encourage continued discussions on this. I think they're really important discussions. And one example that's a very recent example, I just wanted to provide, as we recently approved tovorafenib for pediatric low-grade glioma, and it was based upon a response rate endpoint with durability of response. However, we did include minor responses in addition to the partial and complete responses, which is a deviation from our typical way of assessing response. And in that case, we made that decision in large part because there was supportive evidence kind of demonstrating that these minor responses benefited patients that was provided with the application. And so, this was very important to us to help enhance our understanding of what that meant to patients. And, these kinds of discussions about how to plan for that are very, very helpful when thinking about how a new endpoint, a new way of measuring response might be used.
Thank you, Marta. I think Denise, you are next.
Yes, I just wanted to just bring back a key point as we're talking about what types of data we would bring forward and guidance that you would give. That all designs that we look for going forward really are, forward thinking, probably out of the box of what we've done in the past, but that they can be inclusive of all patients. We have no patients to waste. We can't run the risk that we're losing patients simply because they can't be seen at one or two major centers of excellence. We need to be creative. Those patients can have, validated or verified tumor histology remotely. We live in a world of technology, so we need to figure out ways that we can be inclusive of all patients and harness all data that we want to get.
Yeah, I think it's an important point, Denise. I think before initiating any clinical trial, that's why it's very important to have early dialogue, and that's what Pierre was saying, that some companies sometimes actually compete, or and then we see, maybe some developments, could be done in a different way, but of course we cannot really intervene in that. So I think that's point of collaboration and cooperation, and before actually starting a clinical trial, making sure of what would be the available potential candidates and see what would be the best strategy and rationale for using one. And as you said, Denise, it's also really important for patients that we take what would be the best candidate drug to be tested. And I think that has to be discussed in an open manner with all academics and regulators. Yeah, Sylvia?
Yeah. I have a question for the group here that is looking forward to the next meeting that for sure we organize in fall, where we are already very much busy, but not busy enough. We might think of going a little bit more into the details of the PUSH project that we are trying to apply for collection of data with some more specific example, maybe a disease-oriented. Then my question is, could be worth inviting also one or two or three companies in the room, and with that I mean especially, is there any risk that this is perceived as a conflict of interest for us as a clinicians that are of course clearly working with this company? We cannot develop new treatments and not also repurpose a drug if we do not interact with companies. So the first two workshops were without companies, and this in a way clean the table from the idea that there is a particular interest. For me, the only interest are patients and having drug for them. So if it is applicable, if it is on purpose to have a company or two companies, I don't know. I've not yet anything in mind in particular in the room. Could this work? And maybe this is a bit late, but is there any suggestion that you have about this point, how to engage company also in the repurposing process that is maybe less sexy than trying to develop a drug in a new indication that, even though is rare, is the way to enter a market?
Yeah, I think that, as many of you have said, many of these discussions have to be done with all the partners involved, and we've discussed regulators globally. We've discussed health technology assessment bodies and payers, and absolutely also the industry, I would say. And if there is any concern about finding the right framework, whether it is for a product-specific discussion or for a general discussion, we can certainly find the right framework to do so, to have those discussions. So don't think that that would be a limit. Okay.
So I think in the interest of time, we are a little bit behind. Maybe Sylvia and Pierre, you want to take the closing remarks, the closing words? Thank you. Thank you to all for this very vivid discussion.
So the first remark is one word only, thanks. Even 3 words, thanks a lot. Sincerely speaking, I believe that this was a very productive workshop on top of the first one. I believe that we understand each other better and better, each other meaning the physicians, the patients, and the regulators here. We are all on the same boat. We want drugs, efficient drugs, to be available as soon as possible in acceptable conditions of safety and efficacy to the patients in need for that, even if the disease is an exceptional one. And we must consider the challenges associated with the rarity of the disease not being a good reason for doing nothing, being a good reason for doing much. So we spoke of efforts, and Francesco mentioned the efforts that anyone is performing to make things possible. I believe this is something difficult. If you want to work on easy topics, this one is the wrong one. No, we must work together. It doesn't mean that we will be always in agreement, that we will always conclude that we are on the same line. On the principles, yes. Maybe at the time of a given submission, we will say, no, that is not acceptable to the regulators, and you will say, oh, the regulators are too demanding too much, but okay. However, we will do our best, sincerely, to bring the best to the patients. This is what is important. If we must adapt for that, you and us, we will. No problem. It's not that difficult, I think, from our side. It's more difficult for you because you are bringing the evidence to the regulators, and this is not something which is very easy. We completely acknowledge that. For us as regulators, we just have to demonstrate availability, flexibility, goodwill. Please trust us in the fact that we are ready to accept that. So if we must consider retrospective data, even considering these data as exploratory to build some better evidence, no problem, we are ready for that. If we must consider a new endpoint, which is the clever endpoint that will evidence the benefits to the patients, no problem, we are ready for that. And if we must invent new processes, anything like that, we will do so. We do that regularly during the scientific advice procedures. We do that at the CHMP when accepting some marketing authorization applications that do not meet the classical requirements. We even have legislation for that, which is the legislation under exceptional circumstances. So no problem. We are here to offer advantages whenever possible to help you in recruiting companies that could be convinced in the end by a proper development, a proper evidence, and with the fact that as regulators, we could help them in saying that, no, we won't ask the moon at the time of the marketing authorization. We will be reasonable considering that you will not get any profit from this new indication, but only the glory of being helpful for the patients, which is something, believe me. So I really trust that over the next discussions, whether specific of a product, specific of an endpoint, specific of a process, anything like that, we will find very nice agreements. And because we are exactly on the same line, happy to be back this evening in Paris with the idea that the meeting was far from being for nothing. I'm very, thanks a lot. It was a gift to me. So thanks a lot, my friends.
So, of course, on behalf of EORTC and all the colleagues in the room, I also want to thank you and Francesco and all the other people of the EMA and also FDA for having us with you, for discussing, for opening, and for starting a path that for sure we will use very much. And our responsibility is now to try to put to draft a manuscript where we want to summarize what we said all together during these two workshops. I think this is also very important signal to pharmaceutical company if we want to invite them next meeting to show that we have started this kind of discussion. The second thing is that for sure we will identify a day for the next meeting, and I wonder if we can maybe prepare a little bit more the contents as we are thinking of going a bit more into an example. Especially we can stalk maybe Kit a little bit, trying to prepare everything related to the methodological approach and maybe also the others. If you agree, we will proceed in such a way. And with that, I also want to thank very much the patients who are also colleagues in this on this road, and wish everyone a safe trip back home, especially those who travel a lot to join. Thank you very much.
May I say a few words, actually? So for those of you who don't know me, my name is Jerry Feeney. I'm actually a myxofibrosarcoma patient of several years and 6 recurrences. In fact, I'm expecting another operation in the next few months. But I'd really like to say that I'm encouraged by the motivation and enthusiasm that I've seen here today towards these very worthwhile objectives. And I'd like to thank you all for your efforts and for this opportunity to be here today to join this forum and hopefully to contribute. Thank you.